Okie dokie. Well, welcome everybody. You finally made it through security. That's a good start for the day. We're here for 2 days, and before we actually start the event, I want to thank, first of all the foreign travelers. We have people here from Iceland, China, I think a couple other places, but I've seen them here, so they actually made it through security before the American citizens. So, so I really want to thank Dr. Collins, Dr. Hodes, and Dr. Eliezer Maslia for urging us forward. The ADSP APOE work group has been hard at work on the efforts to put this meeting together for some time. It looks like a jam-packed schedule, and we have 1,001 people registered. Okay, so everybody is going to be having questions, I'm sure. We have start times both days 8 a.m. The shuttle bus is supposed to let you off here, but who knows what happens. And then today we were going to end at 5, but I think we're starting late enough that it might be 5:15 or 5:30 before we end. The end of the day on the second day is 3 or 3:15, so you guys can get yourself on the road. I want to take just a second to thank our NIA staff, Tiffany Rowe, who knows where to find all the stuff I can't find. Amanda, who is going to be our timekeeper, raise your hand. She will be waving fingers at you to let you know how much time you have. We have Miriam Glenn, who has what does she have? The bell. So when you hear the bell, it's time to come back. And we also have Austin Yang, who is on the road right now, on his way in. So for questions and answers, we have, for sessions 1 through 4, we will have questions online and questions in the room, and we'll try to alternate back and forth. And we have our folks from NIA are going to make sure we get as many questions and answers as we possibly can. And then for the brainstorm, we have Slido. So people will be from outside will be asking questions, and Slido prioritizes questions. So if we get a lot of the same question, we'll get those up to the front of the list of questions to answer. For coffee breaks, there's that cowbell. For lunch breaks, there's that cowbell. The lunch boxes will, if you ordered a lunch, will be at the back of the room. There's also the cafeteria. I gave them warning that you guys were coming, so they might actually have enough food for you. And there are also food carts fairly close to the building, but I have to look at them to see where those are. Leaving campus during the meeting is a bad idea because you have to come back through security, and we know how much fun that was. The next question is, where are the restrooms? Answer, turn left or go straight ahead. If you are taking taxi or Uber, you want pick up and let off points at Lincoln Drive, and they can take you from there. I talked to the security people this morning when I was coming in, and they're aware you guys doing that. And so it's time for me to turn this meeting over to the scientists, and first, Dr. Maslia.
Use this forward, backwards.
Thank you. Thank you. All right. Good morning, everybody. Thank you for being here today. It's great to have you back in campus after so long, and it's really very exciting to see all the great things that we're going to talk about today, and the program that we have. Let me see, how do I advance this? How do I advance it? All right, here we go. So I'm going to be very brief because I know we're a little bit behind on the schedule, and there is a lot of great talks coming up, and certainly you guys have a much greater insight in the field than I do. Just to get us started, just want to first thank Marilyn, Michael, and Tiffany for putting together the meeting, and again to you for coming today, and also for all the people that have registered and are online. Thank you for joining us. It's really, as I said, very exciting having this meeting, long overdue. And I just was gonna give a little bit of a brief overview that would open the door for the discussions that we're gonna be having. I think that from the position paper that you guys put out and all the discussions that the work group has been having, it's quite clear that APOE4 have a gain of function, toxic function activity that it's targetable for therapeutic purposes, and I think a good deal of that we're going to be talking about. However, what it should be important to take into consideration is that I think one of the things that we realized after all these years of research and all the work that you guys have done is that certainly APOE is not just like a monofunctional type of molecule, but it's really more multifunctional, and probably is involved in some sort of plasticity and membrane repair through the effects on lipid metabolism and lipid transport at the membrane level. And that could be reflected in a number of different cell types. I think for many years we were focusing on the effects of APOE in one cell type, either glial cells or inflammatory pathways, and then don't work on in neuronal pathways, and then later on oligodendroglial cells and the vasculature. So I think that what we have learned over time is that the effects of APOE, and in particular the toxic effects of APOE4, are not limited to one cell type, but are actually certainly possible in different cell types, and the mechanisms through which each cell type and the signaling pathways through which each cell type will be affected might be different or might be somewhat related, but certainly could be different. For example, like in neurons might be mediated via DLK, MAP kinase, ERK, or in glial cells via Junghah kinase, so TLR4 and NF-kappa B, or in oligodendroglial cells via interactions with APCA1, et cetera. So probably when thinking about both pathogenic mechanisms as well as therapeutic development, given that some of the therapeutic developments that are going to be developed are going to be targeting, let's say, APOE4 in total, well, that is going to have effects on multiple brain regions in multiple cell types, and certainly also in multiple cell compartments. I think that's the other issue, right? Probably while in neuronal cells might have some effects on certain neuronal compartments, endosomal, lysosomal, probably in microglial, astroglial cells might be more relevant what happened in lysosomal pathways or autophagy or in the mitochondria, in the case of oligodendrocytes in myelination and so on. So there is a great complexity when thinking about therapeutic approaches targeting APOE4, or in the upside, if there is a lot of function because we're targeting APOE4, what do we need to do to have some APOE3-like effects that might compensate some of those effects of APOE4? And certainly, I think you have shown a number of different effects on A-beta and tau clearance, as I mentioned, signaling, autophagy, even transcriptional alterations that could lead to the proteinopathy, the inflammation, neurodegeneration, vascular damage, and so on. So that's really the concept that this is more like a polymorphic type of situation where one needs to attack the problem from multiple different angles is very clear. And I think the fact that we have been so lucky in the last several years of having the additional funding for the national plan to address Alzheimer's disease and having a great rich community like you working on APOE from very different angles, I think is really bringing the field together And we're trying to understand finally this elephant that had so many parts that we were trying to look at together, and trying to understand this in a more total, more holistic way, which certainly I think hopefully within the next couple of years, we'll have the first APOE targeting therapeutic out there. Well, with that said, let me just finalize by saying that just in a couple of weeks, we have our NIH 2024 AD Research Summit. This is also very exciting, and certainly the work on APOE will be certainly in mind during the summit and quite relevant, as we will be focusing on precision medicine. And of course, APOE4 is probably one of the best and most clear targets for precision medicine approaches. So this is September 23 and 25 here in at Nature Building 45 here in the NIH campus. And if you can't join us in person, certainly please register and join us online, and we'll have some exciting discussions and results and data, and from there we'll have some exciting revision of the milestones and new milestones that will serve as the basis for what is going to be the next phase of the national plan, because as you all know, the national plan is was for 2025, which we're at the right at the next corner from it. So Congress is in the process of renewing for 2035 the national plan, which of course will be very important for all the work that you're doing and we're doing. So without more comments, thank you so much for being here, and let the games begin.
Some people on Zoom are hearing an echo, so please, if you were to just double check, if you're logged in on Zoom here in the room, make sure that you're muted so that there is no echo. And we are moving to our next talk, which is the keynote lecture from David Holtzman. Thank you.
All right, well, it's great to be here. It's been really fun that Marilyn, after meeting with ADSP and Richard Hodis and Francis Collins, organized an APOE working group, and it's been really fun being with many of you involved in working on this to try to really advance this area forward. This is such an important topic, given that APOE is the most important genetic risk factor for late onset Alzheimer's disease, that we know a lot more about it than we do, surprisingly, after over 30 years of knowing it's a risk factor. As I get started, I first got interested in APOE before many of you, but not all of you in the room, who discovered that APOE was a risk factor. So I started getting interested in this in 1993. When I was still at UCSF, and began working on it then, and things have changed a lot over the years in terms of our knowledge of how this might be acting. So I'll try to go through some of, at least from my perspective, where things are with APOE4 and some of the other factors that might lead ultimately to therapeutics on this topic. Here's my disclosures. Okay, so while this is an oversimplified graph, I think there's an incredible amount of evidence from human data that in terms of the time course of some of the main pathologies of Alzheimer's disease, that the amyloid beta peptide starts to aggregate about 20 years in the brain before symptoms begin. An inflammatory response ensues almost immediately. And then something happens where we don't completely understand how, but A beta downstream drives tauopathy, And that the tauopathy seems to correlate very strongly with decline in cognitive state. There's many other factors, of course, not listed here, but I think one of the big issues in my view is how is it that APOE is influencing, where is APOE influencing this kind of process, and how is it doing that? And it may not be simple, straightforward, it's only one thing, but I think I've always thought about this in the context of the course of disease. And while the amyloid beta cascade hypothesis of as it was first put out was probably markedly oversimplified, I think there is incredible amount of evidence that A beta is necessary but not sufficient for this disease. And I think one of the things that has emerged is that some of the key questions in the field, not just about APOE, but just in general, is how is it that, how is it that A beta somehow is driving tauopathy? How is it that inflammation is interacting with both A beta tau and other factors in the pathogenesis? And where in this process is APOE acting? I think there's a lot of evidence I'll show you that it acts certainly on affecting A beta aggregation, but it also appears to be involved in a number of other processes related to inflammation, tauopathy, and other factors. So I'll try to go through some of those, at least as how I think about this at this point. So we know APOE is the major genetic risk factor for late onset AD. Of course, we know that the E4 allele increases risk relative to E3, and E2 decreases risk. It appears that APOE4 is also a strong risk factor, well, it is definitely a strong risk factor for cerebral amyloid angiopathy and also a risk factor for Lewy body dementia. And that understanding that is not, the understanding of how that is not the case so much. I think a lot more work needs to be done on that. Depending on the population you're looking at, E4 appears to increase risk with one allele relative to E3 by about 3.5 to 4-fold, and 2 copies about 12-fold. Again, you'll hear a lot more about this from the geneticists over the next days. And E2 decreases risk quite a bit. So as we put out in this position paper from data from Jeff Vance's group and others, shows that the risk of E4 being homozygous for E4 is not uniform across ancestries. And I'm not going to spend time going over this, and I'm sure I know others will during this conference. And I think this is going to be very important to understand why this is the case. So there's a lot of things APOE probably does to contribute to AD and related neurodegenerative diseases, but I think there's certainly an incredible amount of evidence that one of the main reasons it plays a role in risk for AD is its effect on amyloid. And let me just go through at high level why that looks to be the case. So if this is the time course of the different pathologies in AD, let's say amyloid and tau, if APOE was affecting amyloid based on isoforms, what one would predict is that in people that are E4 positive in a dose-dependent way, it would shift that red curve to the left. And it would, if you're E2, it would shift it to the right. And that's exactly what you see in people and in animal models. So this is just an example from one paper that we published a number of years ago, and there's other data from Eric Ryman's group and others that if you, this is a group of people, over 200 individuals that we studied at the WashU ADRC who are all cognitively normal, 50 to 70 years old. And we just simply asked, what's the prevalence of amyloid deposition in that age range? And if you focus on the left graph, what you can see is that this is a marker, CSF beta 42 below a certain number marks if you have amyloid deposition or not. So what you can see is that the people that were E4/4, over 60% of them with an average age of 60 had amyloid deposition in their brain. If you had one copy of E4, it's about 35%, E3/3 about 25%, and E2 almost nobody had amyloid deposition at that age. I mean, that's what some of there's a recent paper in Nature Medicine which basically has a lot more patients, but it shows the same data. So one of the things that got me really interested in the fact that APOE, A-beta interactions are important is a paper that came out a number of years ago by Steve Paul and Kelly Bales when they were at Eli Lilly. And they basically took the first amyloid model that was created, the PD-APP mouse, and they simply crossed it to APOE knockout mice. And what they saw was a very dramatic effect. And of course, in these mice, that by about 12 months of age, they have a lot of amyloid deposition, as marked here. When you remove APOE, no amyloid forms. I hope everybody remembers this paper because it tells you that APOE is required for amyloid deposition, which would mean that any form of APOE might be important for this phenotype, not just one E4. Now, there's some A-beta that aggregates in the absence, of APOE, but none of it is fibrillar. None of it is true amyloid. So at this time, my lab had already made the first transgenic mice that expressed human APOE in the brain, and we expressed it under the control of GFAP promoter because most APOE in the brain is made by astrocytes. We crossed the mice to APOE knockout mice. So the only APOE they were making was APOE 2, 3, or 4, and only in the brain. And when we crossed the mice to amyloid mice, what we found is shown here, which is that the mice at about 12 months of age, in the presence of E4, started getting a lot of amyloid beta deposition. With E3, there was quite a bit less. And in the absence of APOE, you saw amyloid A-beta deposition, but none of this was fibrillar. None of it was true amyloid. Subsequently, many groups have seen this type of data from models they've been using. This is just another example where Joe Castellano, then a graduate student in my lab, crossed APOE 2, 3, and 4 knock-in mice. So everything, APOE was controlled by the normal regulatory elements in the genome. And they crossed this, again, to PD-APP mice. And what you could see is by 15 months of age, there was a very striking isoform-dependent effect where E4 had much more A-beta deposition than E3, than E2. And this is very similar to what you would see in humans, basically. So when we looked at the mechanism underlying this, we took the same animals and we looked at how fast the A-beta peptide was cleared from the brain in young mice before there was amyloid deposition. And we used this technique shown here where we did microdialysis where we could measure the A-beta peptide in the interstitial fluid of the brain and then stopped A-beta production with a gamma secretase inhibitor so you could see how fast A-beta was cleared. And what you can see is that the clearance was slowest with E4, less with E3, less with E2, suggesting that one of the mechanisms that APOE leads to this pathology is by affecting the clearance of the soluble form of A-beta. We also, others have found that A-beta also directly affects the aggregation itself of the A-beta peptide. So in regard to these effects on A-beta, would one want to increase or decrease APOE levels from a therapeutic perspective? Well, from a genetic perspective, our lab and Yadong Wang's lab did a very similar experiment. We basically took amyloid depositing mice and we crossed them to mice that express APOE3 or APOE4 with 2 copies of each, or we just crossed the mice so they only had 1 copy of E3 or 1 copy of E4. And you can see this dramatic decrease in A-beta deposition with 1 copy versus 2 copies of either allele. We didn't do this for E2 at the time. I don't know if Yadong has subsequently done that, but I mean, I think this would argue, at least from birth on, lower levels of this protein is probably going to decrease the occurrence of amyloid deposition. And data from Mike Greicius that he'll probably talk about in humans looks like in rare people who only have have a dysfunctional copy of E4 also get very little AD pathology in the few people that have been examined. So one of the ways that we decided to try to see if therapeutically we could lower APOE was this experiment that we did in collaboration with Ionis Pharmaceuticals, where we used an ASO to APOE, human APOE. And first we found that if you just administer this ASO at birth or at a few months of age, after administering it, we could lower APOE levels by about 50% in the brain. This is with a single ICV injection. So we went on to use this same system to take mice that develop amyloid deposition, and in the first set of experiments, we injected, we injected the ASOs right after birth, and that's before amyloid deposition is started. And by doing that, we could find a similar result as the genetic experiments, where basically lowering APOE by about 50%, E3 or E4, lowered amyloid deposition. The other interesting finding is that the number of dystrophic neurites around the amyloid plaques was also lowered. We also did experiments where we injected the ASOs after amyloid deposition began. And when we did this, and again, it lowered we lowered APOE levels by about 50%. When we did this, we saw no major difference in the effect on amyloid deposition. Now, it's possible, and I think you'll hear a talk tomorrow about this, that maybe if the levels were lowered even lower than 50%, we might see an effect in animals that already, or maybe people that already have amyloid deposition, but we didn't see an effect in this model system by lowering it by 50%. Even though we didn't see an effect on amyloid, we still saw an effect on decreasing dystrophic neurites around the plaque. So maybe the toxicity of maybe APOE is also leading to further amyloid-related toxicity, and by lowering the level, maybe that would be an important issue as we talk about therapeutics, especially tomorrow. So where is the APOE coming from that regulates amyloid deposition? I think this experiment, it's maybe a little hard to see, but the bottom line is that if we look at mice, for example, that develop amyloid deposition that express APOE4, and we only remove APOE from astrocytes, you get almost no amyloid deposition. And that's a very similar phenotype as simply removing APOE completely. So I think in regard to that phenotype, most of the APOE that's driving this probably comes from astrocytes. However, other groups have looked at the effect of APOE that comes from other cell sources. So in the top results, Oleg Butovsky's lab removed APOE4 only from microglia in an amyloid model. And his experiments, there wasn't a big effect on amyloid deposition by only lowering APOE in microglia, although the reactive state of the microglia was affected, implying that there's a cell autonomous effect of APOE on the ability of the microglia to become in different inflammatory states. Guojun Bu's lab did the opposite experiment where they took mice that had no APOE and they only expressed APOE in microglia. And there, again, they didn't see a big effect on amyloid deposition, but they did see that APOE had subtle effects, or had cell autonomous effects on microglial function, such as on lipid metabolism and inflammatory gene expression and lysosomal pathways. So I think there's a theme that perhaps there are cell autonomous effects of APOE produced by cells like microglia that might be relevant to some of the phenotypes that we're interested in studying. There's a number of groups who've tried to overexpress potential protective variants of APOE, especially E2. And I'm just showing a more recent paper that came out of Brad Hyman's lab. I think this will be discussed more by Brad during the meeting later. But in these experiments, he used mice that we made that are APP transgenic mice that are E44, and then he overexpressed APOE2 only in ependymal cells in the brain. So the APOE was secreted into the CSF, and then it would get into other parts of the brain. And by doing this, the animals already had amyloid deposition when the experiment started, and the animals that in a dose-dependent way show a decrease in amyloid deposition over time. I would say it's not clear whether this was inhibiting further growth of amyloid deposition versus clearing amyloid, and I think further studies need to be done to address that. One of the things that we know in humans and in animals that APOE4 in particular does is it markedly affects cerebral amyloid angiopathy. And this is a paper we published over almost 25 years ago where we took one of the first amyloid depositing models that Karen Ash had produced called TG2576. This particular model develops not just amyloid plaques, but also cerebral amyloid angiopathy. And when we cross these mice to APOE knockout mice, the mice never develop any cerebral amyloid angiopathy, showing that APOE is required for CAA formation. Subsequently, one of the graduate students at the time in my lab, John Fryer, took these same mice, these TG2576 mice, and crossed them to APOE4 and E3 knock-in mice. And if you look at the picture on the top, what you can see is that in the presence of mouse APOE, you get parenchymal amyloid deposition and A-beta deposition in CAA. If you cross these mice onto an E4-4 knock-in background, most of the A-beta deposition moves into CAA. So the only thing we're doing is changing the presence of E4 versus mouse APOE. And if you express APOE3 instead, there's less A-beta deposition, but most of it is in the parenchyma and not in CAA. And we still don't understand the mechanism of how APOE is leading to this altered parenchymal versus amyloid CAA deposition. I think this is an understudied area that needs to be further worked on. From a therapeutic perspective, our lab found this interesting finding at the time in collaboration with Denali Therapeutics, where we had developed a number of monoclonal antibodies to APOE, and one of them, interestingly, only binds APOE in a conformation that is present in amyloid plaques and CAA. It doesn't bind the normal physiologic form of APOE that's lipidated in our brains and in our bodies. And when we applied this, we gave this antibody to mice that developed both parenchymal plaques and CAA. This is a model, this is an APP model that expresses E44, so they get a lot of CAA. And when we give this antibody peripherally, it actually decreases both the CAA and the parenchymal plaques, kind of like an amyloid beta antibody, except amyloid beta antibodies don't remove the CAA. They just decrease the amyloid plaques. And as you know, many of them cause this side effect called ARIA. So interestingly, this anti-A-beta antibody, in this case, we used a version of aducanumab. You can see that it causes a mouse version of ARIA-E or ARIA-H, which is hemorrhages. And this APOE antibody does not do that. And we're trying to understand the mechanism of why it doesn't cause ARIA, but it's an interesting way to potentially target APOE, only the form that's related to amyloidosis. So what about the other phases of AD? Obviously, one of the big elephants in the room is not just tauopathy, but how molecules like tau lead to damage. And so there's a really interesting paper you're going to hear a lot more about from several talks that was shown just a few years ago in 2019 in this Nature Medicine paper, that there was a rare Colombian kindred subject with a presenilin mutation who did not become cognitively impaired until her early 70s, which is about 25 years after the rest of her kindred becomes impaired. And it was found that she has this APOE3 Christchurch variant of APOE in homozygous state. Now, when this first came out, one, it was a really dramatic single case, but I don't think, to me, it was clear that is the reason that she had this delayed pathology or delayed onset of cognitive decline due to the fact that she's homozygous for this variant or some other reason. So, and this, of course, you've probably seen all of this data before, but in her case, she developed a lot of amyloid deposition, but very, very little tau pathology, which was really striking. It was almost like the pathway between A-beta and tau was being blocked, if that's well, for whatever reason, whether it's the variant or something else. So you'll hear more in a talk later today by Yun Chen, a very talented graduate student now, postdoc, where basically we made knock-in mice for the APOE3 Christchurch variant, crossed them to amyloid mice, and then used a model where you inject Alzheimer's disease tau into the brain and look at amyloid-induced tau seeding and spreading, which is something we think might be analogous to what as occurs in AD. So in this model system, what we find is that in the presence of, after these injections, you can see tau present in dystrophic neurites. It looks very similar to what you see in an Alzheimer brain that's both seeded and spreads to the other side of the brain. But in the presence of this Christchurch variant, this is markedly inhibited. So it's kind of mimicking what you see in the patient. And Yen will go into this in much more detail later today, but around the actual amyloid plaques, you also see an altered microglial phenotype in the presence of the Christchurch variant, where a disease-associated microglial phenotype seems to be amplified. If you try to understand whether or not what the mechanism of what we're seeing with blockade of amyloid going to tau might be, Yin did experiments where he took just isolated myeloid cells to mimic microglia from these mice. And these cells take up tau more readily, and they also degrade it faster. And the myeloid cells then release, even though they take up more tau with the Christchurch variant, they actually release less tau that can seed back into the media. So maybe microglia are playing an important role due to this variant in being able to take up and degrade tau, preventing this connection between amyloid and tau. And that's some of the experiments that we're further pursuing. And one of the mechanisms that many people who've been studying this variant are working on is the fact that the variant doesn't appear to bind to heparan sulfate proteoglycans, one of the co-receptors with LRP1 very effectively. And if we look at the uptake of tau by myeloid cells from the Christchurch variant, what we can see is that they take up tau more so than with E3. But if we apply APOE3 to the cells, it actually inhibits tau uptake, but not so much if you apply the Christchurch variant. So what we think might be going on is that tau and aggregated forms of tau and APOE may be competing for the same receptor systems on different cells, including microglia, and that in the presence of the Christchurch variant, you're actually much better able to take up and degrade tau because you're not competing with that receptor system with APOE. So that's something that we're working hard on trying to better understand, and I know others are trying to do this in different ways. And I'll just show briefly one slide from Yadong Wang's data where instead of using an amyloid-induced tauopathy model, Yadong's lab used the P3NS tau model. And he, like we had found previously, that in the presence of APOE4, you get a lot more damage in the brain in this model. And that if you put the Christchurch variant on an E4 background, you block most of the injury in this model. So there's something about tau and injury that this variant is really having an effect on. And I think that'll be really important to understand mechanistically and perhaps therapeutically. Okay, so Mike Greicius, I'm sure will talk about this, but there's recently other rare protective variants in APOE that have been discovered. One's called the Jacksonville variant, which is B236E on an E3 background. That is shown here. And he also described on an E4 background, the R251G variant. And I think it'll really be important to understand the mechanism of how these rare variants are protective. It could be that it's similar to what's going on with the Christchurch variant, but it could be very different. So I think understanding which aspects of pathogenesis these variants are hitting is going to be important. So several years ago, one of the experiments that our lab did is we just simply asked the question, does APOE affect tauopathy or tau-mediated neurodegeneration? And we used a model, the P3NS or PS19 model that overexpresses the P3NS tau variant. And these animals that Virginia Lee first made many years ago develop a lot of neurodegeneration where tau accumulates in the brain. And this is an example here. You can see the hippocampus is very atrophied where a lot of tau is developing, as well as the entorhinal cortex. And if these mice express APOE4, this damage is much worse than E3 than E2. And perhaps what was most striking, in the absence of APOE, there was almost no injury at all. We've repeated this experiment multiple times. And one of the things that gave us a clue as to what APOE might be doing is that if you look at the inflammatory response in the presence of APOE, you can see that there's a very strong increase in reactive microglia in the presence of tau and APOE4. And in the absence of APOE, this microglial response is virtually blocked. So somehow APOE is affecting this in microglial 3 minutes. Okay, inflammatory response. This effect can be of blocking neurodegeneration, can be mimicked simply by removing microglia from the brain with a small molecule. And that's the same phenotype as you get if you remove APOE. We can also find, and this is not necessarily related to APOE, but just removing the adaptive immune response by removing T cells affects the microglial T cell interaction and is also incredibly neuroprotective. So perhaps understanding how the adaptive immune system interacts with not just the inflammatory system, but also APOE might be an important area to further study. So if we actually lower APOE4 therapeutically in a model of tauopathy, is that something that would be worth pursuing clinically? So we took the same PS19 mice that express APOE4, and again, in collaboration with Ionis, we're able to show that you could lower APOE by more than 50% in the brain with a single injection. And if you do the same experiment and then follow out what happens to the animals, if you don't lower APOE4, you get a lot of damage in the brain. And if you lower APOE4 by about 50% over several months, you can see that there's significant neuroprotection. So I know others that are working on siRNA and APOE ASOs. We'll talk more about this tomorrow. Finally, one of the other ways that one could approach attacking APOE is indirectly by one of the things that happens in glial cells, in particular in damage, is they develop a lot of lipid accumulation, and APOE is probably involved in this. And one way to get rid of lipid from cells is through activating the LXR pathway and ABCA1. So recently we published a paper where we showed if you give an LXR agonist to these tau mice with APOE4 in particular, that it decreases injuries substantially, it decreases tauopathy, it decreases inflammation, both the innate and adaptive immune response. And the large increase in lipid accumulation that's seen in glial cells in these mice is really abrogated by giving the LXR agonist, or what's not shown here, by overexpressing the ABCA1 protein in the brain. We can also lower APOE by overexpressing the LDL receptor in the brain, and that is also neuroprotective. So I think whether we want to increase or decrease APOE in the brain, I think we need a lot more work on this. You'll hear data that overexpression on certain variants might be protective, but certainly lowering APOE in many situations can be very neuroprotective in animal models. I'm going to skip through this and go to the end. So I think it's very clear APOE is the most important genetic risk factor for late onset AD. In regard to the main phenotypes relevant to AD, such as amyloidosis, tauopathy, inflammation, and neurodegeneration, I think there's no question that E4 exerts a toxic gain of function effect. Lowering APOE4 in the brain would be an important therapeutic target for AD, and whether lowering or raising other APOE variants in the brain would be beneficial, injurious, or neutral, I think does require further studies, and I know this will be discussed during the conference. Further studies of protective APOE variants is warranted, as they can have profound effects on different aspects of AD pathology, and APOE can have cell autonomous and non-cell autonomous effects, which may be possible to take advantage of therapeutically. So I think I'm at time, so I'll stop and just show thank you to many people who were involved in these studies over the years.
Push the button. No, that's a thing. Well, no, you'll be able to see it, but what are we doing? Where are we right now? Because I thought there was. That's all right. So. Okay, you can talk from here.
Yeah. Okay.
Yeah, you can talk from here. Yes.
Yeah, I'll introduce you. Oh, so we can see it through there?
Mm-hmm.
Okay, perfect. Seamless. Everything is seamless this morning. All right, it's my pleasure. I'm Jeff Vance at the University of Miami, and we're going to start off by talking about diversity and other data from the ADSP. I'll be moderating, and Allison will be my co-moderator handling questions on the chat. They're coming through chat, right? Yeah. Through chat. So if you're online and you have questions, please put them in the chat. So our first speaker is someone I've just met 47 years ago, I think. And she's going to give us, Dave has given us a really nice molecular history kind of and Peggy's going to tell us about, set us at the very beginning so we can then move ahead. So Peggy.
Hi, and welcome, and thanks for the invite. I'm going to talk a little bit today about the history of discovery, which I like to call the unsung heroes of APOE discovery, and then introduce the topic of diversity, which will be explained in more detail by my co-speakers, Dr. Rajabli and Dr. Griswold. So how did it all begin? Well, I came back from maternity leave, and Dr. Roses, who many of you know out there, said, hey, they're giving money at the National Institutes of Aging, and they're going to give you $7 million for 7 years if you put in this application for a LEAD award, and in Alzheimer's disease. And I said, Alan, he goes, well, Dr. Heyman doesn't want to do it. They want me to do it. And I said, Alan, what do you know about Alzheimer's? He said to me, I saw some patients during residency, and I said, works for me. And that's how we got into Alzheimer's. Now, and then he said, there's one catch. You have to come up with a genetics project. And you can't use those big pedigrees because this guy named Hislop and Haines already have used them, the early onset one. So I said, okay. So I went to see Dr. Heyman, who is a wealth of information. Dr. Heyman, if you know, was important in the CROD, setting up the CROD battery. And so he said, you know, Peggy, I think there's something going on in these individuals between age 65 and 70 or 75, really 60 to 75. And I've been collecting these pedigrees. In fact, I've collected these pedigrees, and I found this researcher named Ann Spence, and Ann Spence does lineage analysis. So I collected blood samples, sent it to her. We ran a bunch of markers, and we have a paper in press looking at these families for linkage. But we didn't find anything because this is right before Gazella came out with his paper on Huntington's disease. He said, in fact, we even looked at heterogeneity and didn't see any. I said, okay, we can work with that. So we started with his families. We put in a paper. I had been a student of Elton, so I put in a paper to do non-parametric, a grant to put in a project into the LEAD award to do non-parametric CRISPR analysis. And Alan was actually funded. And that's how we got into Alzheimer's disease. However, we spent the next couple of years expanding Dr. Heyman's families and looking at our families. And soon I realized that doing SIP pair analysis wasn't going to make it because we didn't have a lot of SIP pairs. We had a lot of avuncular pairs, we had cousin pairs, and I wasn't going to have the power I needed. These are just a subset of these families. So I looked around for different methods, and I found a young graduate student, Dan Weeks, who had written a method called the effective pedigree member method that looked at whole pedigrees. So but nobody really looked at it, nobody was using it, so we decided to use it and said, let's try it. And Ken Lang was his advisor, and what happened was we found chromosome 19 linkage. We found that linkage to 19 and 21, and the reason we found 21 is because the family in there, we had one early onset family that ended up in Alison Goate's original paper on APP. So we found both 19 and 21. We were really excited. So what did we do next? We decided to look for candidate genes in the region. We started looking for genes in the region that had anything to do with brain. But we found nothing. We must have looked at 10 different genes for association and found nothing. And then there was this guy in the lab, Warren Strittmatter, and Warren Strittmatter were looking for things that characterizing proteins that were in amyloid plaques. And my office was next to Rose's. And I heard him and Warren talking one day, and Warren said, we find this thing, apolipoprotein E, but it's only involved in cardiovascular disease. And therefore, I don't know why it's coming up. So at that time, I was head of the chromosome 19 committee for gene mapping, and I knew APOE was in our linkage reason. So we did a quick association of the data we have, and we put it into the PNAS paper, which I don't even think at that time those papers were reviewed, and hence it was published. But it came to fruition that everybody, kind of ignored it until the science paper. And I have to say that we first submitted this paper to Nature. And being young and naive, I didn't know that they sent all those papers to John. And John actually owned up to it. John said he told Nature to reject it. And he actually said it in the editorial in Science. I thought it was a flawed study. So, and so I turned around and sent it to Science. And John Haines called the editor at Science and talked him into sending it for review. So they sent it for review, and it still remains the most cited original research paper in AD. So thank you, Science. So where do we go? This is what we found. We found a dose effect for risk and age of onset. This is from Corder's original paper. Oh, it's not moving. I also wanted to point out that originally in the paper, we happened to say that the homozygosity for APOE4 was virtually sufficient to cause AD, and that was pretty much ignored too, until the Nature Medicine paper that came out recently. It's pretty exciting because they confirmed that the homozygosity, but they also added in additional data of a biomarker in clinical. So we really established this as APOE being a unique finding. Now, the problem is that everything we did, we had looked at families, so then we went ahead and looked at the large series of sporadics that Ann Saunders published. And then I put this in here because, especially for students, as we were at a Gordon conference, Jonathan, Neil, and I, we decided to play hooky and went to the beach in Newport, Rhode Island, and we started talking about APOE and APOE2 and how it was protective, and Neil said, let's prove it. And that's how the paper on protective effects of APOE came out. And this time we did submit it to Nature Genetics, and they accepted it. So now a little bit about diverse. Everything we had done so far, though, were non-Hispanic white populations. So all the results we had initially obviously came out of North Carolina, but then, it's not moving. Why is it not moving on me? It's not working. Can I? Where's our guy? Stuck.
Right.
No, he's got to the summary. No. Well, now I don't have to talk. He just said the whole talk. And it's not moving.
Here, try. No, it is.
Oh, now it is. Okay, thank you. He knows I'm not good at technical stuff. Anyways, the original Porter et al. Paper, and then there was a Japanese report that showed a very high risk in Asian populations. And then Mayeux et al. Looked at African American, Hispanic, and non-Hispanic whites in his publication, added that in. Then Hugh Henry, who started doing with the African American in Indianapolis, and then the reports from Ibadan. So we started getting these reports from other populations. And then there was another report from Hong Chinese that also saw an increased risk in Asian populations. So we looked a little bit, and now I'd like to say the seminal paper by Lindsay, which I think really sewed up, and people still reference it today, where he looked at non-Hispanic whites, African Americans, Hispanics, and Japanese, and put these all together in the largest paper and the largest study done. I remember sitting in a room when we were putting this together. And it still is an important critical paper. Now, recently, Beloy et al. Went ahead and looked at it again and basically didn't find anything that different from what Lindsay found. He has tighter confidence limits, but he basically confirmed the early study. He had 68,000 in his paper, Beloy. So I just do want to make a comment because we have, so now all the Hispanic populations have been grouped together, but through the ADSP, and I'll get done. Through the ADSP, you know, we started looking at these different collections, and we found that there are differences, and to put them together, there is heterogeneity across these populations, and this just gives you an example of some of the publications that have been out there, and with the highest risk factor being seen in the Peruvian population. Now, real quick on Africa, there's been a few studies on Africa, and these are the countries that are involved, but our new looking at the Dawn study, where we're going to be looking at 10 countries in Africa. Our initial data, and this is looking at West Africa and East Africa, shows an increased risk, especially in East Africa for 4 individuals. But as you notice, we have very little data, and the confidence limits are huge. So we need more data on that. This is what we're expecting to come into the Dawn population. Real quick, these are some updated risk factors from different Asian populations, but also shows the high risk in Asian populations. And then if you look at this, I'm going to introduce Dr. Rajabli, who's coming next. And you look and you see the heterogeneity in risk across different ancestral groups, with the Asian population still having the highest risk and the African populations the lowest. And I think Fareed's going to tell us a little bit about why this can be and why this happened. And finally, many reasons lead to Rome. These came together for the discovery of APOE and its contribution to AD risk. The absence of findings can be as important as their presence.
Off the stage. So as Fareed mentioned, I'm going to talk a little bit about some functional follow-up that we've been doing with Jeff Vance and the team there in Miami to try to add a functional layer onto the ancestral risk that Peggy and Fareed were just discussing. And so I'm going to take a particular angle on this and talk about APOE expression. So I'm just showing you here a plot from GTEx that shows you all the tissues and the expression of APOE in them. The 2 largest peaks there are in the liver and in the adrenal glands, and that likely has to do with APOE's function in controlling lipid metabolism. I'm going to ignore those and focus specifically on APOE expression in the brain, and particularly how APOE is regulated depending on ancestral background and AD status. And so GTEx takes a look at this from a big bulk perspective, but there are obviously a lot of single cell analyses that have been done over the last several years that have tried to narrow down from brain to specific cell types in the brain where APOE is the most active. So here I'm showing you a plot from the CAD project. There are 2 clusters up in the upper right that are sort of highly expressing APOE, and these are your microglia and astrocytes from the frontal cortex. You can see that there's some low-level expression across the brain in other cell types, but those are the 2 primary places where we find APOE expressed. And I'm just showing you this now in a second data set. This is from a Mathis et al. Paper that was published just a few weeks ago. This is a huge paper, nearly 2 million cells across dozens of samples across multiple brain regions. And the take home, at least for APOE expression, is still that it's primarily expressed in microglia and astrocytes, some expression in oligodendrocyte type cells and in endothelial cells in the brain. And so knowing which cell types APOE is primarily expressed in, I used this Mathis et al. Browser where you see the link on this page here to look at the difference in expression in APOE depending on neuropathological diagnosis of AD. So they have a really nice browser where you can do this. And so I'm showing you a volcano plot here of differential gene expression in astrocytes in samples where the individuals were neuropathologically diagnosed with AD versus those that were had no neuropathology. And in the red circle here, I'm showing you that APOE is more highly expressed in the AD cases. This is in astrocytes. When you do the same analysis in microglia, you'll see APOE again very up there in the top right. So this does suggest that APOE is more highly expressed in the astrocytes and microglia of individuals with AD, but these were sort of a big cohort. There's a mix of E4s, E3s, unknown APOE. And so we had several research questions that we're looking at the University of Miami that ask whether E3 versus E4 have differential expression. So I'll talk about that. And then how does this relate to what we've just been talking about in terms of risk and ancestry? And so we'll look at that, and then we'll finally talk about some of the underlying mechanisms that, spoiler alert, there are some differences, some of the mechanisms that might be regulating that. And so first, I'm going to show some single cell data. This is from our own work, Dr. Sophia Mora, a postdoc in the lab. And this was just comparing individuals that carry APOE33 or APOE44. And you can see, at least qualitatively here on the right, there's a lot more purple. And primarily, the highest levels of purple are still in the microglia and the astrocytes. But you can see, again, this low level expression of APOE throughout cell types. If you quantify this, it's about a 1.8-fold increase of expression in carriers of E4 compared to E3. So this may be at least one mechanism by which E4 is contributing higher risk, is by this higher expression in these cell types in the brain. And so Fareed mentioned this paper. This was work done by myself and Dr. Katrina Sellis, and this was published a couple of years ago in Alzheimer's and Dementia. You can read it at that QR code. I'm showing you a similar plot here, where again, I've highlighted the astrocytes and the microglia, and here we're comparing only E4 carriers, but E4 carriers that either have European local ancestry on the right or African local ancestry on the left. And you can see, again, there's about almost a 2-fold higher increase in APOE4 expression when the local ancestry is European. And so there's at least an association with higher expression of APOE in the microglia and astrocytes when the risk is higher in Europeans. And so this at least suggests that increased APOE expression is associated with higher risk for AD. We tried to dig a little bit deeper and explain this by looking at chromatin accessibility. So in this same set of samples, we performed single cell ATAC-seq looking at chromatin accessibility, so from these same individuals. And we see that this increase in expression is at least partially explained by an increase in accessibility around the promoter of APOE. So if you look at the right, again, this is looking in single cell ATAC in astrocytes. You can see these sort of two high peaks, much higher in the European local ancestry brains than in the African local ancestry to the left. This paper is also published in Alzheimer's and Dementia last year. So again, the QR code changed even though it didn't look like it. And then to dig a little bit deeper, we wanted to ask the question, are there regulatory elements in the neighborhood of APOE that might be explaining this difference in expression between the European and African local ancestry? And so this is work that was led by Dr. Karen Nuttemens in collaboration with Alison Goate at the University of Pennsylvania. Again, this is published, and you can see this with the QR code here. What we did is we looked across the haplotype of the African local ancestry and the European local ancestry, and we found blocks across the genome where there were differences in allele frequencies between those haplotypes. And that's what's represented by sort of all of these numbered blocks that you see on the browser. And then we performed two different assays. One was an MPRA, a massively parallel reporter assay, where we cloned all of these little fragments onto a reporter vector, put those into cells, and then we asked the question, are these regions capable of driving expression? Are they enhancer-like regions across the genome? And so you can see highlighted here in yellow what were two of our highest signals. So what we're calling block 10 and in block 13. We were able to show this both in the massively parallel reporter assay and then individually in clone luciferase assay. So we have orthogonal confirmation that these regions in this intron of TOM40 have regulatory potential. And then working with Dr. Chessy, what we were able to show in the lower part here through a promoter capture high C analysis is that these regions that have regulatory potentials have chromatin loops back to the APOE promoter. And so this suggests that not only do they have regulatory potential, but that they're likely, but that they're actually controlling the expression of APOE. And so we've taken this one step further. So this is some unpublished brand new work from Dr. Aura Ramirez. And so what we wanted to do is use a CRISPR-based approach to essentially snip out these regulatory regions and then see their effect on APOE. And so she's done this in IPS lines that we then differentiated into microglia. So these have either what we're calling block 10 or block 13 removed by CRISPR. And then we've just measured the expression of APOE. We're continuing to characterize these more, but I think what's a pretty exciting finding is if you look at block 10 and you compare in the African local ancestry, when block 10 is removed by CRISPR, the expression of APOE increases about threefold. And so it looks that this is perhaps an APOE repressor. We're still sort of characterizing that, but it does seem that there are specific regions in that haplotype that are regulating the expression of APOE. And so this is just to conclude what I've just told you, that APOE is highly expressed in the brain, more highly expressed in AD versus controls. APOE4 is more highly expressed than APOE3, that on the European local ancestry, that expression of APOE4 is even higher. That this is in part driven by chromatin differences and regulatory architecture, but that there also seem to be some ancestry-specific mechanisms that are sequence-dependent on those haplotypes. And we're continuing to look at this. Any good talk ends with more questions. And so, you know, it seems like the consensus may be that a reduced expression of APOE4 would be a therapeutic goal, but the questions are, how low should we go? What timing of this should we focus on? Systemic versus local effects in the brain, all of these are, you know, questions to be considered. We're continuing to use other techniques to try to resolve these mechanisms. So we have a lot of chromatin capture going on to look at the structure of the genome. And we're thinking about doing some other high throughput regulatory modulation. And then we continue to grow our single cell data sets in other ancestries, particularly Amerindian and Asian backgrounds, and to think about other genotypes beyond 3 and 4. And I will finish there right on time, I think.
All right, so now we're going to have a period of for questions. We'll then half from the people that are here, and the other half of time if there are questions from the chat. And I think we'll also that for our 4 speakers here, and also for David. So. And I've been instructed to make sure you say who you are and where you're from. And if you can't remember, just look at that badge that we took hours to get. David, do you have a question? Go ahead. Oh, go ahead, David. Oh, just a stand.
Right. Okay. So I wanted to ask a question to Tony. The data you showed was great. One question is, in terms of the single cell analysis of APOE gene expression, I imagine most of the samples you're using had AD. So the pathology itself is going to change the expression of, you know, the gene, because the microglia get activated, they start making more APOE. Have you ever looked at, have you done the same analysis in people of different genotypes before or in the absence of AD pathology to see if the baseline regulation without having a disease process there is affected?
I don't know if this thing. Oh, perfect. Yeah, I don't know if I need it, but yeah, so that's a really good question. So I should say, from our own data, everybody that we looked at are AD cases. So just to avoid kind of that mixture. There, a lot of the other data sets that are out there are mixtures of kind of case control. And so some of that data that I showed on the first couple slides are sort of looking at a diagnosis perspective, right? So I think that leads to exactly what you're saying, that the activation of microglia is going to drive some of these expressions. I think, you know, our focus was really on using the ancestry as the comparator. So we do try to control for, you know, we don't, you know, not all of our Africans are very early stage versus all late stage Europeans, for example. So we have thought about that in the design, but we are now starting to look. I think the idea of looking earlier on is a really important one. And then looking, one of the things that we don't have, but that we're starting to generate now are in unimpaired individuals across the ancestries with single cells. So most of the single cell data sets are all European. There's with maybe a handful of African local ancestry sprinkled in, but we're going to be generating some of that data on our own to do that comparison directly. But
I guess the iPSC astrocytes and microglia can also get at that.
Yeah, so that's something that I in 10 minutes I didn't have time, but we have an entire sort of set of projects looking at those iPSC-derived oligodendrocytes, microglia, astrocytes, neurons, sort of all of those to do those comparisons. But brain versus cells, there's a whole discussion to be had. But yeah, it's a good suggestion.
I will say that one of the problems is that the number of autopsy materials available for non-Europeans is somewhat limited. It really needs to be increased, and we're working hard. Peggy and Goldie, a bird, are working hard to try to get more African Americans involved. And Amerindian is, I can't even find any. So.
Yes, Eduardo Marcora Mansana. I have two questions, one for Anthony, one from Dave. Is this just for you guys, or Dave can also answer?
Dave too.
Dave too.
Dave's not. Dave is a victim as well.
I'll start with Dave then, because I don't have a photographic memory. It's only a few figures that got stuck in my hippocampus, and one is from a paper of his from about 20 years ago, where you showed knocking out APOE in an amyloid deposition model and showing dramatically decreased parenchymal deposition, at least, like Kelly Bell, like you said in the paper, in the presentation. But the next to it was a knockout of APOE lipoprotein J or clustering, showing very similar effects as knocking out APOE. But then next to that was the double knockout, where you saw the opposite phenotype. You saw a much increase, even over wild type, amyloid deposition, even much earlier deposition. So my question is about this kind of framing of thinking about relationship between APOE or other lipoproteins and amyloid deposition, just in linear terms, gain of function, loss of function. You think that's still useful, or maybe hindering our understanding of what's really going on?
I think it's still useful to think about it this way. The issue, though, is that there's probably more, APOJ may have similar effects as APOE does in regard to affecting A beta metabolism and clearance and aggregation. So there's more than one molecule that affects a similar process. You may need to understand the complexity of what regulates, for example, clearance of a molecule. And I think it's knocking down only one thing might be relevant therapeutically in understanding, but it may be complicated if you start modifying other things at the same time.
A quick question for Anthony. So
probably just one question at a time here. Dale, you can ask him at the break. Julie.
Julia TCW from Boston University. I have a question to Barrett. So there was very interesting result that, the other reported that African American has carrying E4 lowering like 56% of compared to that of European background. But when you actually compare, it was actually surprised that East African has a ratio of over 19, which is really stronger than European. And have you compared East African versus West African or African American in terms of local haplotype or APOE expression? Why those are very East Africans are much stronger risk?
Yeah, very interesting question. So I just need to note that the data we showed about the West Africa and East Africa, it's just recent data we collected in last month. We did all this analysis. For your comment on the E44 effect in East Africa, where we observed around 19.5, keep in mind the confidence interval is very wide. And we have just, I think, around 12 individuals carrier of the 44, and most of them are AD. And this is the clinical, from the clinical data. So I think we still need more data to have a good idea what is would be the effect in East Africa. And on the other side, when we look at the East Africa E34, which is has a much higher frequency of E34, we actually didn't see the even significance.
But
so,
I do, go ahead. But I do think, how did you do that? How did you make the red?
I think you're on.
You're on.
Yeah.
Oh, I'm on? Okay.
Yep.
All right. And so I do think that the patterns emerging, though, that 4/4s have exceeding risk across different populations. So, and where we see the real differences in terms of risk is in the 3/4s. I mean, yes, the confidence limits are high, but we're starting to see a pattern there. We see the pattern in, I think, in Peru, in the Mirror Indian. So I think over the next couple of years, and we'll see we'll be able to sort some of that out.
Yes, and about your question about the West Africa, if we compared, in West Africa, it was really a very quick talk, so I couldn't talk too much about. We actually were trying to look at in West Africa in different sites. So we have around 6 sites in West Africa. And when we look at the Yoruba, which is previously studied with African Americans, and in the previous studies, they couldn't see the significance in E34. We couldn't actually see significance as well in Yoruba, which is almost 100% having an African ancestry background. When we added the Ghana and the other sites, which having some admixture from other sites like Middle East and in Europe, we start to see the significance. So it's kind of still we see that the African background having a protective effect.
Hi, I am Mia Sung from the Intramural NIA, and I want to thank the organizers for this very educational workshop. I'm learning so much. I have a quick question for the last speaker. So when you showed the CRISPR knockout of the enhancer that seems to reside in the intron of the neighboring gene, since the chromatin landscape is depending very, thank you. Yeah. Since the enhancer landscape depends on the tissue and the cell type, I was wondering, I might have missed this. Which cell type did you do the experiments on?
So those were done in both iPS-derived microglia and astrocytes. So in both cell types, we saw those differences.
I see, and the landscapes were pretty similar between astrocytes.
It's similar. So in that region, I'll say yes, it's similar. That region interacts with the APOE promoter in those 2 cell types.
So that particular enhancer apparently was important for both cell types.
Correct.
I see. Thank you.
Thanks.
Front. Hi, this is Michael Haney from UPenn. I had a question for Tony, kind of in that same vein. Really great talk. I was wondering if this regulatory element you think it's unique to regulating APOE expression in microglia and astrocytes as opposed to the periphery. I asked this from like a kind of wondering from a therapeutic standpoint if this is lowering APOE levels throughout the entire body and if that has detrimental effects on other organ systems or predisposes to other diseases.
That's a really good question that I do not know the answer to. The title of my talk was in brain cell types. So that's, so again, just to keep, the enhancer regions that we identified are from looking at frequency differences in alleles between the 2 haplotypes, right? So in principle, that the sequence changes exist in every cell type. What I don't think I've looked at, and I'm not sure how much data is out there, is, sort of the promoter to enhancer chromatin effects, whether that's as I just said, whether that's a cell-specific thing or whether if I looked into hepatocytes or something, if I would see those same interactions. I don't know that answer, but it's a good question worth thinking about.
I think later we'll have a talk on comparing peripheral and CNS.
Thank you.
You don't?
Yeah, Yalong Huang from Glassland UCSF. A question also for Tony. So when you compare the expression in astrocytes or microglia across different ancestry, did you look also the different subcluster of those cell types? Because you are working on the single cell data, right? So, for example, those DAM or ARM, those are microglia. Did you see difference across different populations? And also, if so, what about the APOE expression across those different subcluster of those cell types?
Yeah, we looked at this in the original paper where we were comparing the European and African backgrounds. And so what we did notice is that particularly in Europeans, there's a cluster of microglia, of astrocytes that look like they're sort of activated astrocytes. They have the highest expression of APOE, and it's a really very obvious cluster that we see. And so we did that. In terms of the different classes of microglia, given the cell numbers that we had, we weren't really able to get down into that granular of a detail from the brain tissues that we looked at. But I think it's a really good question, and one that as sample sizes increase, that will become an easier thing to do when the cell numbers are big enough to make those comparisons.
Thank you. Do we have questions from the virtual chat?
So some of the people who've got up and answered questions will take the one. All right. Take Gil question, and then we'll go to the ones that are up there.
Go ahead.
Hi, I'm Gil Di-Paolo from Denali Therapeutics. So regarding the ancestry-specific effects of the APOE genotype, there's I'm sure you thought about this a lot. Modifiers as well. TLC gamma 2 has been reported. There are others that will be discussed at this conference. But regarding the possibility that there may be common variants that are protective or deleterious that are enriched with generation-specific ancestries, is this also a scenario that you're envisaging for to explain those changes in the impact of the APOE genotype? And if so, have you done some work to document that?
Well, you didn't present the data for the common. One of you two can.
For the what?
For the common variant you found.
Oh, well, I'm going to talk about that in a minute.
You're going to talk about it.
But I would say that the studies that Pericak did and others, it's really the local ancestry. That area seems to be the key point for the ancestral differences. They looked at, globally at other genes, but in an environment indirectly, and it doesn't seem to contribute to that difference.
But you have found stuff, but you'll present in the local ancestry in the Haptoglobin area.
Yeah, in the local ancestry, I'm going to talk about a protective effect.
I'll be patient.
I think patience is the key word this morning. Do we have questions on the?
Yes, we do. So
why don't you use the microphone?
All right. So a couple of questions related to age and ancestry and risk. First, was asking about Asian populations beyond Japanese and how does it compare across different Asian populations. And secondly, there was a question pointing out that age and lifespan are actually much higher in Japanese populations, and is that controlled for in the analysis? You want to start?
Yeah, we actually observed a higher risk, and where in our first slide we showed that we see that in Korean population and Chinese population the higher risk in A34 allele versus A33 in these populations. There's we see that there's the highest risk in Japanese, but it's the confidence intervals are kind of there overlaps. About the controlling for the, I haven't done those analyses. I don't know if they're controlling those analyses, but yeah, that's something important to consider.
So, and I also want to point out that I think that so many more studies need to be done. I think this is like the tip of the iceberg. I know the stuff that like Li Wang and the ACAD consortium that I think Richard is involved in, where they're actually recruiting 5,000 Asians of different U.S.-based Asians across the U.S. I think that's going to really help. I mean, it was just a smattering of results, and I think we'll start seeing the same things we see in the Hispanic population. There's a lot of heterogeneity and variability. We just don't have the numbers yet to predict them.
Then a question I think directed to Tony. So a couple of things. One is, does APOE RNA expression correlate with APOE protein levels across the different isoforms? It says anonymous attendee actually, so maybe. And then I think sort of following on from the question Dave mentioned, and I see sort of not asked in quite the same way, but also from the chat, is, you know, are the differences in APOE isoform expression present at basal levels, or is it related to some environmental induction related to Alzheimer's disease that produces a magnified effect of E3 versus E4?
Yeah, I'll answer the second one first, which is, I think that's something we need to find out. I think we don't know that answer, whether this is without getting too cliche, whether it's a chicken and egg problem, whether this is a disease response or a disease causing. I think that the genetics suggest that there's a real cause here, but I think that's still something that we're working on. The first question related to protein, I think that we can safely say that APOE protein is made in astrocytes and in microglia. I do not have the answer as to whether 3s and 4s and by ancestry the protein levels change. You know, the effect is really localized to those two cell types, and so when we, if you look at bulk RNA, for example, that signal is sort of muted out, and it's hard to find. So we really need, you know, we have some of the resources available in IPS models across ancestries to begin to ask that question, but we've not done that yet.
Genetic question now, but Peggy. So even in Europeans, there's a gradient of risk from north to south, right? So do we have any idea, this may also be true for the ancestry too, which is how much of that might be explained by local genetics versus environment? Sorry. Okay. Did everyone hear my question?
Yes.
Okay.
I heard it.
Yeah. So when we know that there's differences in the effect size, Southern European and the North European populations. We also actually assess the risk in our Puerto Ricans when we stratified by their European background and check their risk with the Southern, the risk that has been published in Iberian Peninsula. We observe the similarity. So it looks like actually those risk effects have been inherited in the mixed populations, which is correlates with genetic ancestry. Is there any effect of environmental factors? It's always something we need to consider. But when we looking this ancestral difference, we're looking within the same population. So we're looking at the ancestral difference within the same population and stratify them by the ancestral backgrounds. By looking within the same population, actually trying to control the environmental factors. We see this African versus European. We don't have really any population. We have like a mix of the Southern European or the Northern European to have this to better assess this effect. But
so how much has been looked at actually across all of these ancestries about the haplotypes that exist across ancestries? And because it seems from the data that local ancestry, so therefore local haplotypes, is likely to be important, and so how much variation is there in the haplotypes that E4 sits on, for example, across populations?
Well, I certainly think we can infer that there's a big difference in haplotypes just based on the regulatory and the expression differences as well. We're just starting long read, and I think long read will really be helpful here. And we're in the midst of doing long read on different ancestry, so I think that'll be really useful.
Yeah, there was a question in the chat about how many SNPs sort of are along for the ride, so are in the haplotype with APOE that could potentially be explaining these expression differences, and you highlighted these two particular blocks, but how many SNPs are there, and could there be haplotype effects rather than individual SNP effects? Because obviously the MPRA test is one at a time, right? Right. But actually there could be sort of interactive effects.
I can tell you from the High-C data and from virtual capture High-C we're doing, there's a lot of interactions with a promoter, a lot. And so I think APOE has a lot of control, and which makes sense. It seems to be a very important molecule for stress and various other things. So, I think there is a haplotype effect, and that will be really interesting. And there may not be just one. I think there are a lot of different enhancers, and APOE is most highly expressed if you look at Allen Brain Bank right after birth. And so we haven't even discussed that yet, but timing is, right. But that's, everyone, when I say that, they all go, so who knows when different elements are controlling APOE expression.
Yeah, so I see another question from the chat. Maybe it's for Dave, but is there evidence that APOE4 primes astrocytes and microglia to be chronically reactive even in the earlier stages of AD pathology? So anyone on the panel or Dave, do you think that there's a difference in reactivity of E4 astrocytes and microglia in response to damage there?
Yeah, so I'll let you guys, from your data with Julia and others, I mean, you've addressed that to some extent in cultured iPSC-induced cells, but in vivo, at least in animal model systems that only express, for example, E2, 3, or 4, early in life, the astrocytes do not appear, and microglia do not appear to be phenotypically or gene expression different. That doesn't mean that there's not something that we're not seeing, but and in humans, I don't know.
One point is, and I was going to talk to you about this later, is that, E4 is the highest frequency in equatorial regions in Africa and South America, and it's thought because it has a protective immune function against parasites and some bacteria. So, there clearly is some difference there in the immune and that there's not microglia, but macrophages probably. And I think it's time for a coffee break. So we'll be breaking, we're almost on time, and we're coming back at, what time? At 10:35. So thank you all the speakers, and
I also thought that the APOE is highly expressed on that. So it's been hard to characterize that there is in the outcome data we're starting to look at basically bio and extracting methylation data from there. So I'm looking at now not just but also methylation changes. That data but this is the thing, really, we really struggle with visibility of the material because about how to slow it down again, right? So at first, just having another first year now I've got one brain, right? So we have a lot of approaches that is we now have to generate a lot of micro data from the brain as well. So yeah, I'd love for you to get about this one, thing and that's all. Yeah, so we get this question of modifiers when we brought up here, right? So I think that this is something that we think about. If people have really found very clear examples of, African American specific people in the they absolutely exist, right? And people are talk about one of their causes that we might so I think at the end of the day, it's driven by this observation that, the Pericak, which is regardless of anything the rest of the you have be a one negative African area there. So we really focus a lot of effort in that area, and then we kind of go out there. Thank you. Appreciate it. Yeah, thank you. How are you? Wasn't that a I know that's wrong. Right. What he was yeah, I know. Yeah, so is it like a thing?
Okay.
So how do you?
This is forward.
Okay.
That's back. The bottom one is back. And the laser pointer is press and hold only at the screen.
Wow, that's
don't point it at that screen, because if you point it at that screen, only us in the room can see. But if you point it at this screen, the people online can see.
That's so cool. Yeah, I love it. I love it.
I think most of them are like that now.
Oh.
All right.
Thank you very much.
You're welcome. That's what I'm here for.
So I guess I can take that.
Yeah, you can go ahead and take it.
Okay.
You're welcome.
What do you think of this? Right, yeah. In your cohort. I think that we should good method quantify the gap. Yeah. Yeah, because yeah. Yeah. Yeah, you exactly, right. Yeah, so I guess I want
Hi, Dr. Kampmann. How are you, sir?
Doing well, great. Did the screen sharing work?
Yeah, we could see it. If you could go ahead and bring it back up.
All right.
And you could just leave it up since you're the first speaker.
Great, I'll just leave it up then.
Okay.
Oh.
Did you have any videos or anything in your presentation?
No videos, no.
Okay. No problem. Yeah, you could just leave it up, sir, since you're the first speaker.
Great. And you can hear me okay?
Could you increase the volume on your microphone?
Let me see if I can do that. Yes.
Okay, we're good.
Okay. I try to speak up.
Yeah, you sound clear, sir.
Thank you.
Okay, I'm only supposed to be the chat person, but in the absence of Jeff, I don't know. I don't see. Is Jeff coming? Okay. So our first speaker in the continuation of this session one is Martin Kampmann from UCSF, and title of his talk is Dissecting APOE Biology by CRISPR-based Functional Genomics. So sorry you're not with us, Martin, but good to see you online.
Thank you, Alison. Good to see you. I'm sorry not to be there in person either, but excited about this symposium and to share with you some pretty early stage findings, but also to present a system that I think hopefully has potential for some of the questions that we're all interested in. Basically, my lab is interested in using unbiased CRISPR-based screening approaches to uncover mechanisms in neurodegenerative diseases. CRISPR technology is obviously well known to this audience. When I was a postdoc with Jonathan Weissman, we developed some twists on that technology that allowed us to use catalytically dead Cas9 fused to transcriptional activator or repressor domains. We call this CRISPR-A and CRISPR-I, and to do genome-wide gain and loss of function screens that way in human cells, originally cancer cell lines. And then I spent the first years of my own lab combining that technology with human iPSC technology to be able to do genetic modifier screens in human neurons, microglia, and astrocytes. And what I'll tell you about today is how we can do this in neuron glia co-culture systems and 3D co-culture systems to start to get at interactions between different cell types. And this system was set up in the lab by a great graduate student, Emily. What I'll tell you about today, most of it is in a bioRxiv preprint paper as well that we recently updated. In order to study neuron glia interactions, of course, we could use cerebral organoid systems that are sophisticated systems that are very popular to study neural development. But in this case, because we are interested more in neurodegeneration and studying cell type interactions in that way, we decided to go with a somewhat simpler system that gives us, however, a lot of control over cell type composition and so on, in which we separately differentiate iPSCs into neurons, astrocytes, and microglia, and assemble these in agrival plates into 3D spheres. And we obtain a very nice spheroid where we can control cell types, both in terms of their ratios, but for example, also in terms of their genotypes. And we characterized this by single-cell RNA sequencing and found, for example, focusing here on the neurons, that the neurons of equivalent ages in these what we call I-assembloids are much more mature in terms of the expression, for example, of various ion channel subunits and other things related to neural maturation. And also when we look at the activity, for example, by multi-electrode array, we find them to be much more active. So this was then a system for us to start to look into APOE biology. And as the first experiments that I show you today, we decided to look at the impact of APOE, the E3 versus E4 isoforms in astrocytes that are co-cultured with neurons and looking at the effect on neurons. The rationale being that APOE is, of course, highly expressed in astrocytes. We heard from Dave Holtzman that astrocyte-derived APOE in mouse models plays an important role. So we decided to ask the question of what the impact might be on neuronal function and to uncover that using a modifier screen. So the way we did this is to make a guide RNA library against about 2,300 genes representing all kinases, the druggable genome, and so on, and introducing that into our iPSC neurons. In this experiments, we kept the neurons always in an APOE3 background because we wanted to really look at the cell non-autonomous effect, if you will, that APOE from astrocytes would have on these neurons. And so then we did two parallel experiments, co-culturing these neurons either with APOE3 astrocytes or APOE4 astrocytes in these 3D co-cultures or I-assembloids, and collecting samples at different time points such that we could investigate which genetic perturbations in neurons would have an impact on neuronal survival in these two different environments. And we did this experiment using actually two pairs of genetic backgrounds. In one case, we used the COLF 2.1 background, which the NIH INDIE project has started to engineer a lot of AD-relevant genetic variants into, and so we had APOE3 and 4 from that, and we made neurons in astrocytes. We also had a separate genetic background, neurons in a WTC11 background in astrocytes, in isogenic background that was generated by Julia TCW when she was in Alison Goate's lab and generously shared with us. The modifier screens that we did in parallel in these two systems showed that, broadly speaking, there was actually a lot of agreement between hits affecting neuronal survival independently of the APOE3 versus APOE4 background. That wasn't particularly true in our system that used the WTC11 and TCW system. We found a few more differential things in the COLF 2.1 system. We wanted to look for things that were consistently found between the two systems, and at first glance there were not too many things that were had the same differential effect of E3 and E4 in both sets of genetic backgrounds, but there were a few, and they fell into a few interesting pathways, including notably things related to either neuronal activity, synaptic transmission, and metabolism. Here's an example of a gene that we validated and that was interesting to us. This gene's CACNB4 is a subunit of a voltage-gated calcium channel. It was interesting to us because genetically in humans, loss of function of this protein is linked to epilepsy, suggesting that loss of function might lead to hyperexcitability. And obviously, this was a loss of function screen, so that would be maybe the expected phenotype. And so we validated that knocking down this gene in a E3 system was actually not toxic to neurons. So the value of 1 here says that survival of neurons is the same as for non-targeting control guide RNAs, but knocking it down in neurons, and these are the same E3 neurons, but in the presence of E4 astrocytes, was somewhat toxic. So that was interesting and led us to ask whether maybe there is a difference in activity that makes these neurons particularly vulnerable to this perturbation in an E4 glial environment. We did single-cell RNA sequencing on these neuron astrocyte co-cultures and found that, interestingly, two processes, again, were differential in neurons co-cultures with E3 versus E4 astrocytes. In particular, things related to mitochondrial energy metabolism and also neuronal development. And again, looking at this here, we see that those related genes are more highly expressed in neurons when they are in the presence of E4 astrocytes. We also then wanted to see whether functionally there was a difference, and indeed, using multi-electrode arrays, we found again that neurons in the presence of E4 astrocytes had a substantially higher degree of neuronal activity. Also, we looked at reactive oxygen species as a byproduct of mitochondrial, higher mitochondrial metabolism, and there found quite a substantial increase in reactive oxygen species in neurons measured by a indicated dye here, cell rocks, compared to neurons in the presence of E3 astrocytes. This was very interesting, and to my knowledge, hadn't been seen in that way before, although intriguingly, there was a older paper from Li-Hui Tsai's lab that had investigated the effect of APOE genotype in microglia co-cultured with neurons, and they had found that microglia that were APOE4 when co-cultured with neurons seemed to lead to higher neuronal activity or excitability compared to APOE3 microglia co-cultured with neurons. Although that might not be the same mechanism, and this is something we are still investigating. How does this link back now to the original screen that looked at survival of neurons? In this case, we have characterized in our system that indeed it is the relatively high neuronal activity in these eye assembloids that leads to ROS. So for example, if we block neuronal activity with tetrodotoxin here, we also lower ROS levels in the neurons. And these ROS levels lead to downstream consequences such as lipid peroxidation, which we can block with ferrostatin, a ferroptosis inhibitor, but also again by downregulating neuronal activity. And this has an impact of survival. So both ferrostatin and which blocks ferroptosis, and also inhibiting neuronal activity improves neuronal survival actually in these eye assembloid models. And in fact, when we titrate in astrocytes at different ratios in this model, on the one hand, and looking at neuronal activity of a 3D neuron model only, we have high activity, hyperexcitability, I would say, and then increasing the amount of astrocytes that we titrate in, we normalize that a little bit and have less of that, suggesting the astrocytes have an important buffering role. And similarly, ROS levels in neurons go down with higher astrocyte levels. So the idea could be then that astrocytes play an important role in buffering neuronal activity and preventing this hyperexcitability that goes hand in hand with increases in metabolism and ROS species, and that based on the synthetic lethality screen, this hyperexcitability is a vulnerability of neurons, in particular in the APOE4 astrocyte co-culture, presumably because there's something about the APOE4 astrocytes that does not fully have that buffering function anymore. Very early studies, so definitely much more mechanistic work to do. And lastly, I also briefly want to point out that, of course, the iPSC system is a very reductionist system, but to get at more of the complexity, we've also, over the last couple of years, really tried to optimize mouse in vivo brain screens where we can target both neurons and, for example, astrocytes. And an earlier version of this is on bioRxiv, but we still made a lot of tweaks to really get highly reproducible hits, in this case, neuronal survival, but we are also interested in other readouts, of course, in individual mice, screening thousands of genes in a single mouse. So hopefully we'll also investigate those questions in a more physiological context. So with that, big thanks to our lab, Julia TCW and Alison Goate for sharing cell lines, and happy to take any questions.
All right, thank you. Any questions from the group that's present? Yes. Name and location.
It's on, sir. It was on. Yeah, both of them work.
Hi. Martin, this is Gail from Denali. Very nice talk, as always. I
I had a question regarding the DROS and ferroptosis. I read your previous work on prosopagine loss of function causing increased ferroptosis and neuronal death through this pathway, and I was wondering whether there was some sort of convergence with the APOE4 model here. Do you think that the phenotypes that you see could be in part driven by prosopagine loss of function, or is this something that you have addressed? Which, of course, would point to lysosome dysfunction as a key aspect of this, but yeah, just curious.
Thank you, Gil, for that suggestion. It's something that in this model we have not looked at. Prosopagine was also not in the more focused library, but as we go genome-wide, we'll be looking out for those connections with the lysosomes. I would say, as an aside, that intriguingly, after we had this initial paper on prosopagine triggering lipid accumulation, ROS, and ferroptosis in neurons, we have found prosopagine a lot in other genome-wide modifier screens that we have done, including for tau aggregation, where prosopagine seems to make things worse. Yeah, I wouldn't be surprised if it would also exacerbate other ROS-related phenotypes here. But it's a great thing we have to still address directly.
Okay, thank you.
Thank you. We have one question from online.
Yeah, from the chat. So, Martin, this attendee noted that COLF 2.1 and WTC11 are both male lines, and whereas AD is more gender biased and affects more females. And so, does your group have plans to validate your studies in female-derived cell lines? Have you already done it?
We have not already done it, but it's something we absolutely want to do, right? I do think it's very important to validate these things in a number of backgrounds. Of course, also, there's always differences between any two iPSC lines based on genetic background. So we probably have to have quite a number of both male and female lines to really confidently look at sex as a variable here. But it's an exciting thing we want to do. And of course, the mouse model also will allow us to do that in a way that, in that case, takes into account not only the genomic differences, but also other physiological differences, maybe between male and female animals.
All right, thank you. Lihai? Yeah.
Li Wei Cai from MIT. Martin, great talk. I really enjoyed it. But before I ask my question, I just want to offer you too. So we have several isogenic APOE3/4 lines that are from female subjects. So you're very welcome to use them. So your neuron astrocyte co-culture system and hyperactivity and ROS for apoptosis phenotype, I wonder whether the physical presence of astrocytes is really necessary, or the conditioned media from your astrocyte culture is sufficient to cause this neuronal phenotype?
That's absolutely an experiment we need to do, and I think if I had to take a guess, I do think in this case the buffering function of astrocyte, for example, buffering glutamate concentrations and so on, might play an important role. But we have to test this directly to answer your questions, and we're trying to really get at those mechanisms. It's relatively early work, so thank you for that suggestion, and also for offering the cell lines. I think I might follow up with you on that.
Okay, great, thanks.
Yeah, we also have isogenic cell lines, 3 and 4s, in 3 different ancestries as well. So, you don't. Is that on?
Yadong Huang from Glassline UCSF. Martin, great talk. Just a quick question. After you do this co-culture, you analyze the neuron, response. Have you also done on this astrocyte side? So after a few days together, whether this astrocyte also start to show difference, and then those difference maybe respond to neuron effect?
Yeah, there are differences between the astrocytes as well, and we're trying to find clues for mechanisms there as well. I think the other experiment that maybe you're also getting at that would be very interesting, is, given all your work on APOE action in neurons, would be to also switch the genotype in the neurons, right? And then ask, for example, whether APOE in 3 versus 4 in neurons A affects this phenotype and B has an impact back on astrocytes. So those are things that we haven't done yet, but that this very modular system makes easy to ask.
Yeah, that's exactly my next question. Thank you. You already answered.
I think we had. Hi, this is Ring Chen from Washington University. So I'm not sure if you are aware of Shane Lidlow's paper published in 2021, where he actually showed the astrocyte conditioned media can cause neuron toxicity. I don't recall whether she had done with APOE 3 and 4. My first question is, in your system, do you actually see any overlapping response phenotype, these kind of things? And the second question is, he actually showed that the lipid species carried by the APOE is quite important to induce that toxicity. So in your case, does any APOE4 specific lipid carries that can cause this effect?
This is a great question. And yes, Shane has done beautiful work to show that in his paradigm of neurotoxic reactive astrocytes, it seems to be very long chain saturated fatty acids in the conditioned media that are at least accounting for some of that effect. And we have actually, in a separate set of experiments, also tried to recapitulate this in our iPSC-derived neurons. We similarly have found that if you trigger reactive astrocytes and add conditioned media to neurons, under certain conditions, we can measure toxicity, and removing lipids from that is beneficial. We have not yet looked at E4 versus E3, and to my knowledge, neither has Shane. So I think that's a great suggestion to look at this. In his case, however, he does not necessarily see that this would trigger hyperexcitability, hyperactivity in neurons, or be mediated via ROS, to my best knowledge. So I think these might be two parallel mechanisms. And I actually believe that maybe what we're here looking at is not a gain of toxic function of the astrocytes, but more a loss of a protective function of the astrocytes, given that actually when you leave out astrocytes from the eye assembloids, actually some of these things become worse, the ROS and the hyperexcitability. So it might be, these might be two parallel mechanisms.
Yeah, thank you.
All right, I think we need to move on to the next speaker. Thank you very much, Martin. Next speaker is Sharon Kardia from the University of Michigan, who will be speaking about LASI-DAD.
I'm so glad you're doing this and not me. So I actually started working with APOE in Charlie Singh's lab in the late 80s, using genetic data that Eric Boerwinkle. Oh, geez.
You don't have to use it, but that's forward, that's backwards.
Okay, high tech here. Anyway, Eric Boerwinkle, when he was a graduate student in Charlie's lab, had generated APOE genotypes for the Rochester Family Heart Study using 2D gels, and I inherited that as a graduate student. And I have to admit that when Peggy's paper came out on APOE, we thought, oh, it's just linkage disequilibrium, it's nothing. And then another one came out, and we were like, oh, we're way wrong. So I was happy to sort of come back to this in the later part of my career, but one of the things that struck me, and I was wondering, why am I in this session right after a CRISPR gene, is that APOE is an extraordinary gene. We don't have a ton of genes like this. It has such a huge effect on sort of the metabolic processes of not just one gene, but entire systems. And as I will show you here, we've got a little bit of I'll call it the same kind of thing that we're seeing at the population level that you're seeing at the molecular level. And that's, you know, a very sort of I'll call it odd symmetry. So the longitudinal aging study of India was basically built as another large study of aging in India using many of the same principles and survey tools, and even in our case, the diagnostic assessment of dementia based on the health and retirement study. So this longitudinal approach to looking at aging and many of its social, economic, you could add nutrition, there are many, many different sort of layers of sort of rich, you know, kind of data that we're putting together with, for instance, good measures of cognitive data. The LASI study is 70,000 people, and they're following them. The LASI-DAD study is a subsample of about 4,000. We have whole genome sequencing on about 2,700 of them. And there's a way in which this sort of LASI-DAD approach to looking sort of deeply in will, I think, hopefully get us to the place where we can start to look at some of this heterogeneity in a much more sort of rigorous fashion. Many of us know that the population of India is not just vast, but also highly complex. There is a lot of genetic structure, a lot of that having to do with the social structures and basically assortative mating based on, you know, where you are in a particular caste or scheduled tribe or many other sort of elements of the reproductive landscape that actually makes India a kind of hotbed for long stretches of homozygosity and sort of founder effects that are much more magnified than anything that we've seen in the European landscape. Another thing is that they have a discrete, I'll call it cline from north to south of ancestral North Indian ancestry, which we can use as an ancestral background. Given that this is just 10 minutes, I don't have really actually time for that, but there's also a way in which the social structure and the social determinants of health have a major impact. And that's kind of the piece I'm gonna go through, but just know we're also looking at the ancestry differences. We've found modification by age, sex, education, and location, and we're trying to figure out more and more what is behind that. Is it some other environmental, air pollution, diet? There are many different things that are likely to be at play. One of the things that was curious to us when we first started this work was to look at the geographic distribution. As I said, we were looking at ancestral North Indian as a cline, but when we found that there's actually pretty significant differences in the variation of just the APOE4 carrier status, that was more of an indication that we've got much more microgenetic heterogeneity than we had anticipated. And that micro heterogeneity was likely to be not just a confounder, but then also pointing to other things that might be influencing what's going on within these individuals. This is clearly too much to sort of see, but even just looking at the sample characteristics in this group of individuals, we found gene environment correlations that we did not expect. Like I never expected APOE4 carrier status to be associated with education, with residence, with whether or not they were in a scheduled tribe or income. So this again speaks to the structural aspect. So I'm not saying that there's some kind of phenotype relationship, but I do know that it's happening within the social structures. And so it definitely puts an analytical strain on 2,700. So we are, moving to, ask for more samples in the LASI-DAD for its next wave. And then I think one of the big things that's on our radar is to go to the 70,000, because without going to the 70,000, we'll never be able to really actually look within some of these substructures. Another thing that was a big surprise was the amount of variation in the cognitive function measures that we were using. Again, these are based on the health and retirement studies sort of measures so that we could compare across nations. We have a Hindi mental state exam. So it actually is not just Hindi. It was basically translated into something like 15 to 20 different languages. So there's a much more sort of matching to the actual geographic area. We're working with sort of local people who are doing this interview in order to even make sure that there's reduced variation there. Age and sex, the very first column, somewhere between sort of 8 and 20%, not unusual for age to have a huge impact. But state and genetic PCs and putting those together really actually pushed the amount of variation that we were seeing explained, state being a huge predictor of people's responses is another sort of indication that we've got variability here, that we're not exactly sure what is actually tagging along. If we add education, literacy, and rural, urban sort of location, we're in some places explaining 40 to 60%. I've never seen any model of a complex trait where I can explain up to 60% with these basic variables. So there's a lot of variability in here, and I'll be honest, I did not actually think, given we explained this, that APOE would do anything. But I was wrong, and APOE does indeed, on top of this, explain variation in most of the cognitive measures that we had, even when we sort of put in a basic model versus the one that has some of the social determinants. So APOE is going strong, even when there's all these other major sort of predictors of the variability. When we looked within, given that we had some indications that there were going to be modifiers of this, clearly we would expect actually a age effect to be interacting so that the carriers have a greater decline with age. We would expect that. Given that there is a difference between males and females in general with respect to Alzheimer's and other cognitive function measures, we see that as well. And we see a sort of stark effect on education here. We do not see the APOE effect when we're looking at the individuals who are educated. So that tertiary, that upper secondary and vocational even, we're sort of seeing maybe a little bit, but certainly not the tertiary. So this is actually one of the things that is, you know, I'll call it a multi-dimensional interaction. So that no schooling lane is actually mostly women, right? So in this particular culture, there is again these frequency sort of correlations that actually make this very difficult to peel apart without there being very large sample sizes. With this kind of in mind, we have been sort of considering how are we going to bring our team together. So Priya Morjani, who does most of the population genetics work, has done the ancestry estimation as well as looking at the archaic, so the Neanderthal and the Denisovan mapping of fragments onto people's genomes within the whole genome sequence data that we have. Andrea Gana, who is pushing us to do proteomics. We have some methylation work that we're also doing to try to sort of pull together the different levels along with sort of measuring 80 biomarkers so that we can start to fill in some of the gaps.
Thank you very much. Good timing. Questions? I should say that a lot of the sequencing data from LASI-DAD is coming into the ADSP, so it'll be available for everyone. And the effective education, it's nice to see that the effective education that we see in other populations holds true here. And I know Fareed has, Fareed has looked at this in the Amish as well. I mean, in the African Americans, yes, and seen an effect of APOE4 as well. David.
Dave Holtzman, Washington University. Very nice presentation. In terms of what I can't tell from the way you presented it are you separating out when you look at the analysis people who are of that large number of people who clinically who has dementia versus those that don't, and then which biomarkers are you going to use to determine who actually has pathology of at least AD pathology or not?
That's a really good question. So right now, the LASI-DAD team has been working with sort of the physicians to do the AD diagnosis on the whole cohort. And they, they're using, I'll call it a combination of approaches to do that because it's not as straightforward. And this is actually much more of what I think of as a local population study. That 70,000 was originally meant to be representative. So the way in which they looked at the distribution of who to sample means that a lot of people actually have no access to healthcare. They're living in rural areas. And so bringing those individuals into a clinic or bringing the clinic to them has been a major challenge. For the individuals in LASI-DAD, they are going to do sort of MRI scans. They're doing sort of biomarkers, the tau, and but there's not an easy answer. We have a good estimate working with Alden Gross at Hopkins that we got about 7% of the LASI-DAD sample with dementia. But even there, you can have a whole day seminar where we're arguing about just whether or not that's a true number. Is it higher or is it lower?
We have some questions from online.
Sharon, thank you. Sort of several questions around the structure within the population that you sort of alluded to in collinearity of many of these phenotypes, which obviously make it difficult. So a comment about that, and also about the microheterogeneity. So one of the comments says 2,000 recognized ethnic groups across India. Could exploring E4 carrier status by state be revealing the microheterogeneity caused by ethnic customs such as marriage within ethnicities and the caste system?
I think that's right on. I mean, this is one of the least well-powered studies for that kind of question. It's why I brought up that if we can get to the place where we can do it in the 70,000, we would be able to actually answer that with, I think, some better sort of definitive answers. I do think from my experience that it is representing these complex social interactions where even in a location, you'll get what we would call genetic drift, right? I mean, this is something that we would not, think would be unusual if we're talking about hundreds of years of sort of, I'll call it reproductive selective mating in that assortative mating based on customs. And then when you start to add things like, well, then customs go with diet and then with education. I mean, all of the things that are completely confounded in human, I'll call it daily life. And in many ways, when I was looking at the last presentation with those long lists of genes that are basically highly correlated but slightly different, we're likely to find the same thing at the human level with all these social and behavioral determinants of health with the genetic heterogeneity by its side. So it's going to be an analytical conundrum.
Yes, I think that is, I think it's true. And so I was wondering, so I think, how much so you showed quite a big variation in the frequency of people. How much variation was there in the odds ratio? And I think it was on the slide, but I missed it.
So we have it by state, which I didn't include. There are huge differences by state in the APOE effect. And that was the first clue to me that we have something completely different going on in these areas. And while we may only have 250, people in each of these, 250 is pretty good to get an actual estimate that is significant or not significant at that power to sort of point to the directions. So, especially after we've adjusted for variation. So there's a lot of heterogeneity going on within this particular nation.
And just lastly, given your background in cardiovascular disease, how much variability, have you looked at lipid levels in this population, and how much lipid levels changing across these different populations?
So we do see differences in the lipid levels, in particular cholesterol, which we would expect in triglycerides and those kinds of things, but certainly actually not to the same extent as the cognitive measures.
One last question for Reed. You had a question?
Related to the previous question, so when you say that you observe the association between APOE and education,
Fareed, can you get close to the microphone there?
When you mention you observe the association between the APOE and education, did you see the association of the frequency of APOE and education, or it's modified the effect of APOE with the cognitive function?
So I think the question is that whether or not we are looking at the frequency of education or actually the APOE effects on the cognitive measures in the different education strata.
Yes.
It was in the different education strata. Where we see the APOE effects most significantly are in those individuals with no education.
Do you have a biomarker data?
So I think that's being collected right now. We've got it on a limited set of about 500, so we've not touched it because it needs to be actually on the whole set in order for us to have even basic power.
Yeah, this is interesting because we observed a similar pattern in African Americans, and actually when we look at the biomarker data, we observed the similar amount of the biomarkers, the p-tau in this strata, but there was kind of like a cognitive disorder was happening because of medication. That's why that was found.
Thank you.
All right, we'll move on. Thank you very much. Our next speaker is Lindsay Farr, who will talk about, I believe, some Asian data. Is that right?
Good morning. I have the dubious distinction of having findings reported in an ADSP science report a few years ago that were reviewed and scrutinized by Francis Collins, who considered them incongruent, which is code for they don't make any sense. And in fact, it was those findings that led to subsequent discussions of a small group of ADSP members who are in the room today that then led to a white paper led by David Holtzman that then spawned this meeting. So either I can take credit or blame. So I'm going to talk today about that study and as well as some additional follow-up. So as everyone has heard, the effect of APOE4 is highly variable across ancestral populations, with the highest odds ratio among East Asians compared to people of European ancestry, and much lower in African Americans or African background. And the basis for this difference is not entirely clear. We've heard some data today. There are several hypotheses related to modifiers, either within or extant to the APOE locus, as well as moderating influences of dietary and environmental factors, as we just heard in Sharon's talk. So our goal was to look for potential modifiers in the APOE gene, including the regulatory regions. And the study was conducted in a group of Koreans who are participants in the Gwangju Alzheimer's and Related Dementia Study, or GARD, which is directed by my colleague, Dr. Kun-Ho Lee and Jung-Soo Kim, and all of these subjects are part of a whole genome sequence study that is funded by NIA and part of the ADSP. So for this study, we sought out SNPs in the APOE region, and we screened roughly 5.7 kb within and surrounding APOE for common SNPs, and that netted about 57. And then we reduced that number such that they had to be of an allele frequency greater than 1% in 3 ethnic groups: East Asians, European ancestry, African ancestry. And then we further used a criterion to wrong button. I don't know. Hit another criterion where there had to be either increasing or decreasing frequency in the order of East Asian, European, African. And finally, there had to be SNPs with at least 5% difference between groups. So after applying those, we came down to 4 SNPs, and one of those happened to be the E4 SNP. So then that led us to 3 SNPs. And if you look at the data here, the one that stood out to us as showing the kind of variation that we wanted to follow was this SNP RS4055909. So first, looking at the association of E4 with AD by population group, you can see this is already confirming what we've discussed today, that the highest risk is in East Asians, and for us, this was a Korean and Japanese data set, followed by ADGC data sets of European and African American, and the difference in the odds ratio is much more accentuated in those with the 4-4 genotype. So next, we looked at the effect of the variance in this APOE SNP. And by the way, this is a promoter, a SNP in the APOE promoter minus 219. So it was a SNP that was already known. And what we observed is that in the East Asian and European ancestry groups, those that had the T allele had the effect on the odds of AD among 3-4 and particularly among 4-4 were much higher in those with the TT homozygote, next the GT, and the referent group being GG. Here, the numbers, when you cross-match the GG genotype and the 4-4 in the Asians, we didn't have, but the same pattern is observed in the European ancestry. We didn't do this in the African Americans because the E4 the GG, the G allele frequency was much smaller, and once we started stratifying by genotype, there weren't sufficient numbers. So the effect of this allele also influenced age at onset. So in the East Asian groups, those with the T allele had, the G allele had significantly greater chance of developing AD at a younger age compared to those with the GG, and the same pattern of cross genotypes for this SNP was observed in those of European ancestry. Next, we performed a series of experiments, first in reporter constructs, looking at the effect of the T and G alleles on APOE expression, and this was done 2 ways using CRISPR-Cas. First, starting with the T and and editing in a G, or starting with the G and editing in a T. And in either case, what was observed is that the T allele was associated with reduced expression of APOE. And this is the conundrum. This was what Francis picked up. Why would the higher risk allele be associated with reduced expression? It didn't make sense. But just to point out that when we looked at this in both human cortex and cerebellum, we saw the same effects, but importantly, and this is a key thing, the effect of the reduced expression of the T was among those who were only E3 and those who had E4. So it's not specific to the E4 genotype. So subsequent to that study, we did some further bioinformatics work, pulling data out of GTEx, looking at across tissues the effect on APOE expression of this SNP. And what you're seeing is the relative expression of the G allele, the referent being the T. So here you see increased expression of the G allele in cortex, cerebellum, whole blood, but also point out that in other tissues, the pattern is reversed. So there's a lot of tissue-specific expression. And most recently, we looked at this in a context of AD using expression data from ROSMAP brain, so dorsolateral prefrontal cortex tissue. And what we observed is that in a total sample non-stratified by APOE, there was a borderline significant effect in AD cases only of the T allele. So here it's increased expression of the T in AD cases, but this was driven much more so among those who didn't have E4. And interestingly, when we compared across controls, and in controls, by the way, we didn't see any evidence of this, but just comparing the difference in the effect on expression, it was, again, borderline significant. So again, another set of findings to ponder. So that leads me now to what does this all mean? So just posing the question that Francis did, how can ostensibly incongruent findings of the effect of the T allele of this APOE promoter SNP that increases AD risk among E4 homozygotes, but lowers APOE expression? How can you reconcile that? So one possible explanation, and I'm sure there are more, is that the T allele reduces APOE expression regardless of APOE genotype, and its modifying effect on the APOE4-AD association is not through expression, but based on some other mechanism. And that idea is supported by the more recent data that I showed, that the T allele was associated with increased APOE expression in brain of AD cases, but not in control. So this is in the quote unquote right direction. And also this pattern seemed to be independent of APOE4 status. So I'm gonna leave it at that. And I'd like to acknowledge my colleagues at BU, my colleagues from Chosen University, and my ADGC collaborators, I think all of whom are here with us today. And I'm open to taking questions.
Thank you, Lindsay. Still a little confused, but that's my nature. Questions? Vivian? Oh. Yes, Michael.
Yes, Lindsay. This is Michael Benani, NIA. I'm very puzzled by the increased risk in the APOE for heterozygotes versus homozygotes, where the homozygotes have much more heterogeneity in the increased risk when you look at the confidence intervals, if you have an explanation.
Why the E4 homozygotes have a much greater confidence interval?
Yes, than
Oh.
the APOE3.
Yeah, well, that's a largely driven by sample size. The E44 group is much smaller, and especially when you slice by the genotypes of the promoter SNP, the effect is so strong that it's significant, but the confidence interval is very wide. Did I answer your question, Michael?
Yeah, you have. Thank you.
Very nice talk. Quick question. So
Can you
when look at
make
this,
make sure the microphone is right?
when you look at the effect on APOE expression, the T allele seems decrease. We have a look at different type of cells. Maybe across different type of cells in the brain may be regulated differently, even in the same contact of these SNP.
Yep. Can I go back? Yes, that's exactly what has been observed, that the variation in expression, the effect of this allele on expression is not only varies across tissues, but even crosses in opposite directions.
Right, because it's an increase seems in AD. In the AD, there's different type of cells increase or decrease.
Oh, so we have not looked within AD cases in a cell type specific manner, but that's a great thing to do.
Yeah,
Thank you.
That would be very useful to look at that. Vivian.
Vivian Chung from University of Michigan. So it's we've been looking at this region between Tom40 and APOE, including the SNP, very extensively. I'm an RNA biologist. So it turns out that there is an enhancer RNA.
Make sure you have that microphone.
Yeah,
up.
an enhancer RNA in this that includes the SNP. So essentially, at least it could be we don't think that this is a promoter region of APOE, rather that we have very clear data showing that it's a separate TAP RNA.
And can you clarify what the impact of what is the outcome when you're looking at the RNA? Is it AD risk or expression?
Yeah, so we isolated that RNA,
Mm-hmm.
and it acts exactly like an enhancer, right? It touches and promotes the expression of APOE. I probably can't stand here and describe how it works. It's really quite complicated. But I think the transcription pattern of that eRNA, and we can talk offline,
Keep the microphone in front of you there.
explains why there's controversy, because I think what we see is that it is very cell type specific in terms of enhancer function and in terms of in response to stress.
This is the R loop?
Yeah, so this is an eRNA, as Jeff said. It forms an R loop, and that R loop is modified, and the RNA in the R loop is modified and regulates the enhancer itself, then regulates APOE. But we found exactly as you've shown here with GPAS, right, how it works. And it's really a classic modifier because it modifies the expression of the major gene.
Yeah, so the problem that we still have is the link between the effect of the allele on AD risk and what's the mechanism underlying it. The presumption would be, since it's a promoter SNP, is that it's on expression, but I'm not so sure. And if it is, it would, as you suggest, the answer might be sought looking at cell types specific.
Yeah, we can talk. I don't think it's a promoter.
One of the things she stated, Lindsay, was she doesn't think it's actually on the promoter.
Oh, the
SNP. In her studies.
Yes, but so the question is the SNP tagging your RNA?
Yes, exactly.
Yeah.
That's
Yes. Vivian had a nice paper. I think Watts is the first author.
Yeah, and we have a follow-up paper that describes how this RNA works.
Yeah.
Thank you.
R-loop.
Great comment. I appreciate it.
Yeah, it's really interesting. Maybe that's the explanation. We have one question.
Yeah, so, going to APOE protein level data, plasma, CSF, or brain homogenate related to the modifier. Thank you. Sorry about that. I'm a slow learner. So just in case you didn't hear it the first time, is there any data on APOE protein levels in plasma, CSF, or brain homogenates related to the modifier SNP? And I wanted to ask you also can you clarify what the LD is between this SNP and isoform SNP? Because it seems that it's present on both backgrounds, right?
Correct. It's not in high LD. But to answer your question, or the question from the participant on Zoom, is at the present time, I don't know, but we do have data, particularly in the Framingham study, that we would be able to look at that. So that's also a great idea.
Yes.
This is Vincent from Washington University. So I'm not quite an RNA person, more like a protein person. So regardless of those arguments about the RNA, at least you have the data to show that the protein level has been reduced associated with this T-SNP. I guess my question is, regardless, we all AD people, we think APOE.
Make sure you up against the microphone.
Okay, we're pretty sure that APOE4 is a gain of toxicity, but think about that, the human still need this protein, right? So is this level of reduction becomes another detrimental effect because it lacks APOE, sufficient APOE4 in the brain that can be linked to the AD?
I don't think I'm in a position to answer that question, but I like the idea.
So one of the hints can be, do those patients with this T-SNP, do they have any other CNS-related disease risk or these kind of things?
These data are based on screening large aggregate samples. So I don't have any data at this time, but it's certainly
So they don't have any other phenotype associated.
No. Well, again, the reality is as you know, Jeff, the ADGC data, we have limited amount of phenotype data that cross everyone. So of course we could subset and look at some of the data that we have, but we have not done that yet.
Okay, yeah.
Great, thank you. I think we'll move on. We are actually on time, amazing. And our next presenter.
So we're moving into session 2, right? Looks like it's you, is that right?
Is, no. Oh, I guess it is me.
Yeah.
Hold it, I'm confused. Is it you? I mean, no.
No, I'm in.
Oh, yeah, that's the moderator. Oh. Well, the next speaker is absolutely fantastic speaker who has no idea what he's doing. Novel effect, yeah. Any other questions? Stumped them. All right. Well, then we'll move on.
Yep. So the next speaker is Richard Mayeux from Columbia University, and the title of his talk is Fibronectin 1 and APOE4.
So the next few talks, I think, will be on other protective alleles and factors affecting APOE4. And of course, the other thing, of course, is the PSGs, that region, it's all they're mainly expressed in placenta, which always brings up the question of timing of APOE. And there's a lot of data to suggest that APOE is most highly expressed right after birth.
Thank you so much.
It's present from conception. So the timing is always the question that at some point we're going to have to address in our therapeutic approaches. Go ahead.
Okay.
So, Dr. Mayeux.
It's actually Mayeux, not Mayhew. Thank you. There's no H there. It's French.
That should be
okay. So, well, first of all, I want to thank whoever organized this for inviting us to talk. And I'm going to break my discussion into two parts. One is how we got to this point, and then I'm going to talk a little bit about unpublished data and where we are in the second phase of this. I am talking on behalf of work done by Shahan Kazil, who's in the audience, and Badri Vardajian, who's back at home. I hope he's watching. So the concept for this was that we started to identify a number of people who were APOE4 homozygous who did not develop Alzheimer's disease. First, it was in our Dominican population, and then later on, it was in the Washington Heights population. And these were people that we were following up. We could see no change in their cognitive performance. And in some of them, we actually did CSF biomarkers and showed that they actually had normal levels. So as time went on, we collected more and more of these individuals in these cohorts. And then we started to see them in the family-based study that I coordinate with several institutions across the United States. And we decided to look, we had a large enough sample size that we did whole genome sequencing and looked for, separated into two groups. Individuals who were above 80 and had indications of, by more than one clinical visit, of having no evidence of dementia, and they were APOE4 heterozygotes and over 80. And then individuals who were 75 or older and were APOE4 homozygotes and were unaffected, and that we had information suggesting that they were not affected. So in our first pass in 3,500 individuals, we came up with about 80 million variants. We filtered it, insisting that people who were either heterozygote or homozygote and unaffected could have a variant that was not present in cases that were APOE4 heterozygote or homozygote. And that filtered us down to about 510 variants. And then we looked at pathways. And the pathway that kept coming up was the extracellular matrix pathway. And that was very intriguing to us. And two genes stood out, a variant in Fibronectin 1 and a variant in collagen 6. The one in Fibronectin 1 actually was present in all of our cohorts, the three cohorts that we had. So we decided to focus our efforts on that. And we knew a little bit about fibronectin's work. On the bottom, this part here, you can see the allele frequency, and you can see that the variants were not present in the affected people with Alzheimer's disease. So we posted this initial paper on bioRxiv, and then one of my, can I say this, Michael? One of my former students at Columbia, who went on to great fame at Stanford, actually did the second part of this, in which he went into the ADSP data and called us after we had posted our paper and said he was able to replicate the finding. And we were pleasantly surprised, as you might guess. Michael and his group then joined us on the initial paper that showed that a variant in Fibronectin 1 might protect against Alzheimer's disease, both in this large meta-analysis that was totally independent of what we had done, plus the cohorts that we had. So then we decided to look in brain to see whether we could see what role a fibronectin might play. And I'm showing the, in this slide, you can see the increase in fibronectin 1. It's probably easier to see it on the lower panel by APOE4 genotype. And you can see it in the graphic display there. So this led us to hypothesize a couple of things. One is when we compared the E3 cases to the E4 cases with the fibronectin variant, we saw some similarities. One is there was less amyloid aggregation, there was less basement membrane thickening, there was a more prominent microglial response and better clearance, and less synaptic degeneration when we compared E3 to E4. And this led us to hypothesize that fibronectin actually might protect against APOE4 in Alzheimer's disease. But what we didn't understand was what was the molecular mechanism underlying this protection, and could we possibly find a therapeutic compound that would actually mimic the protective effect. So now I'm going to shift into part 2, which is really, a lot of it's unpublished, but it started off with this publication. So one thing we learned from working with our colleagues at Mayo Clinic in Jacksonville was that fibronectin deposition is part of the gliovascular transcription in the blood-brain barrier in Alzheimer's disease. And its main regulatory function, at least at that time we thought, was to maintain VEGF expression. And in, as you can see in the lower panel, in the wild type, this is VEGF, so there's no and this is in zebrafish model. If we then add A beta 42 into the wild type, you see VEGF disappears, but in the fibronectin knockout, we restore the effect of VEGF. Then we looked at APOE4 deposition, APOE4 increased deposition in at the blood-brain barrier and APOE humanized mice that we were able to look at our institution. And you can see there that fibronectin increases in the APOE humanized mice, and you can see it in the graphic on that side as well. And this is both in APP knock-in mice and zebrafish amyloidosis model. And the main effect, and here is where the struggle became very difficult, and that was trying to find brain tissue from people who were APOE4 homozygous and unaffected. And so I called every neuropathologist that I had ever known, and with mostly cold calls. And it turns out that everybody has one or two of these cases in their brain bank. And we were able to get 18 brains of individuals who were unaffected clinically and had either BRCA1 or BRCA2 or BRCA0 and no evidence of cerebral amyloid angiopathy, and could look at those brains. And as you can see, in comparison, fibronectin seems to accumulate in patients with Alzheimer's disease in a higher BRCA stage. There's a sort of increasing in these individuals with the thickening of fibronectin around the blood vessels and near the blood-brain barrier. So this led us to postulate, and we're still working on this, but some functional studies, based on functional studies in mice and zebrafish, that fibronectin regulates the blood-brain barrier integrity through VEGF-A dependent insulin growth factor and heparin binding EGF-like growth factor. And the scenario, it's a cascade, and the scenario goes something like this, that fibronectin loss of function variant upregulates VEGF, so it allows it to be restored, and it affects the expression via the fusion adhesion kinase inhibition. And VEGF inhibition, which happens in Alzheimer's disease, reduces the vascular integrity and also increases or inhibits heparin binding EGF in the endothelial. So the loss of function variant in fibronectin, again, restores the VEGF effect and targets genes in the endothelial, including heparin binding EGF and insulin growth factor-like expression in astrocytes. So in human brains, IGF and VEGF are reduced and fibronectin is generally increased, whereas in fibronectin variant, that is not the case. And you can see this illustrated on this slide. This is unpublished data, but it's it's a paper that's under review right now. So the next step we thought was, could we find a compound, a therapeutic compound that could mimic the effect? And we screened about 270,000 compounds at a facility at Columbia and found 8 compounds that we are currently evaluating. And we validated it in APOE expressing astrocytes from mice. And we found that they were not binding to the active fibronectin in blood. So there's 2 types of fibronectin. There's a soluble form and an insoluble form. And the insoluble form used to be called cold soluble fibronectin. It's mostly in plasma, whereas the insoluble form is really focused at the ECM, the extracellular matrix. And this is just very, very preliminary, but it suggests that one of the compounds, I'll just call it number 3 for now, in APOE4 humanized astrocytes from mice, it does seem to be able to reduce normalized fibronectin integrity, whereas we don't really touch the soluble form. So this kind of leads us to think that, antagonizing fibronectin deposition and the matrix assembly, or at least enhancing its degradation, could serve as a potential therapeutic target. And that's where we are right now. And I just want to also thank all the people that worked with us on this, especially Badri and Shahan. It's a huge effort. But I also want to point out 2 things. One is this work would not have been possible without having the ADSP, because Michael Greicius took advantage of that and was able to replicate it very quickly. And also, I have to thank all of my colleagues at various institutions who put up with my phone calls to ask them if they could find an APOE4 brain without Alzheimer's disease in their freezers, and they all came to us. So this wonderful network allowed us to proceed as we have. So I will stop there, and I'm happy to answer any questions that you might have.
Questions? Julie.
Hello, hi. Julia TCW from Boston University. So actually, you mentioned earlier about collagen 6 as well as the fibronectin. I would like to hear more about the collagen, because we have identified in Alzheimer's brain, they're like a matrixome called matrixome composing ECM factors are enriched, and one of the main core matrices that we found is like collagen, and I would like to know more about that, and is there any association with the fibronectin? And another thing another question is like, what's the reason behind why you specifically look at vasculature? Because it could be also you probably briefly mentioned the astrocyte involvement with fibronectin, and if you ever look at other stuff.
Well, I'll answer the first question first, and that was because fibronectin was present, the variant was present in all of the cohorts. We just decided to go with that one first. The collagen 6 was not. But there are a list of other genes in the extracellular matrix, some of whom I mentioned there, that need to be looked at as well. And in the pathway that we identified, the cascade on one of the earlier slides, I don't know if I can go backwards. Yeah. This pathway that I identified, actually, if you can rescue the effect of the fibronectin, if you can rescue it by affecting VEGF or heparin binding protein. We haven't done the similar experiments in collagen 6, but those need to be done as well. And it may be that this is just an easy one that came first, but it was just present in all the cohorts, so we decided this was more likely to be real than not.
And the second one is that whether you have looked at astrocyte or other whole brain of other than vascular.
We just started with the astrocytes. This is a work in progress, and that's underway. We went first from the zebrafish and then into the mouse, so we've got that working. But other cell types should be examined. Thank you for the question.
Hi, I'm Taka Kanekiya from Mayo Clinic, Jacksonville. So my question is, I assume the extracellular matrix might influence the CAA formation, cerebral amyloid angiopathy. I wonder.
I didn't understand the question.
Cerebral amyloid angiopathy, CAA.
Yeah.
I assume fibronectin mutant variant might influence the CAA severity.
Yes.
Maybe. Have you ever?
I haven't had it. We haven't looked at that, but it's a definite thought because.
Could you repeat the question for the online?
He asked whether fibronectin might affect the severity of cerebral amyloid angiopathy, and I think that's a very good point, but we haven't really looked at it yet, and certainly since it's around the vessels, it might play a significant role.
Do you think fibronectin variant influence a small vessel disease risk in addition to AD?
Yeah, that's a good question. We're very interested in small vessel disease. As you know, about 30% of all Alzheimer patients at autopsy have small vessel disease. Some of it's hemorrhage, some of it's microhemorrhage, some of it's just ischemic. And we don't know if it's a similar mechanism, but it certainly is a viable candidate to be looked at.
Hi, Michael Haney from UPenn. Great talk. I had a quick question on the mechanism of buildup of fibronectin and other ECM proteins in disease. Do you think this could be due to an impairment of clearance from microglia or astrocytes as opposed to just production? And then also therapeutically, I think enhancing clearance from these glial cells as opposed to production would be another approach to remove this excess.
I think the increase in fibronectin, and there's actually some other papers from traumatic brain injury and in radiation exposure in brain, it increases the fibronectin in the same region. So I think it may be a reaction to an external process. Maybe the accumulation of amyloid may increase the fibronectin, and that I think prohibits clearance. So I think I think amyloid does come first. It then triggers this response and then impairs further clearance. I think that may be the way it is. And in fact, in the radiation exposure, people have seen the same thing, that there's an increase in fibronectin at the blood-brain barrier, and they're talking about improving it by increasing degradation or modifying somehow the accumulation of fibronectin at that level.
Thank you.
All right, we'll take one from the internet.
So, have you checked whether the protein expression levels of this fibronectin variant and wild type are comparable? Is it specifically the mutant variant that offers protection against D4 status, or are levels of the mutant affected, and that it's this expression?
I don't think we've done that yet. Have we done? Let me ask Shahan. Have we done that yet?
Correct. Yes. So is it because you've shown lots of function variants, right? Lots of function in your model is protective. Is it the effect of the variant? Is it actually on just modifying expression levels?
Right. So
of the stability of fibronectin.
We are now generating the animal models for the specific variant that may answer this question in a way, but in humans, it's extremely hard to get the tissue and do that. So In humans, we don't have the tissues to really look at that. But what we believe is not the expression levels, but the specific conformation changes in fibronectin. If that may help.
So there's one more question from online, which is, in late onset AD, is the fibronectin-related BBB dysfunction one of the initial causes, like initiating a separate pathway from the amyloid beta tau pathway, or a downstream effect caused by amyloid beta?
I think I answered that already when we think it's amyloid that triggers this response.
Hi, this is Rinchen from Washington University. I'm very familiar with this, and but in my opinion, I have a little bit like loss between the protective variants with your verification of loss of function. So have you ever done any biochemical assessment? This protective variant or the detrimental variant, do that simple mutation will cause any biochemical or biophysical feature change of the insoluble form versus soluble form of the fibronectin itself?
I think that was in the last slide, right? So we just looked at it in astrocytes, right? Humanized.
Yeah, but this is with the compound to reduce it. So I want to ask whether that's specific.
I can answer that. So we have done in silico analysis looking at the grids and the specific structures. The fibronectin has several different domains.
Yes.
The protective variant is fibronectin 2 domain, which is a very hinge region that changes the stiffness. So fibronectin is formed in a certain way and then gets activated by binding to integrins and so on. And this activation requires conformational changes, which we believe is affected by this variant. So we are at the level of the biochemical assays. The interaction of the soluble form and the insoluble form with its binding partners is probably not affected. So we still need to show
we can discuss that later.
Yeah, sure.
I totally know what you're talking about, yeah. I have a question.
Yeah, I think we can have one more question.
Okay, sorry, I wasn't. Nice talk, Richard. It's Gil again from Denali. Maybe you addressed this already and I missed it, but do you have a sense of what cell type in the CNS expresses the protective variant, I suppose? So you do your functional studies in astrocytes. Does it suggest that that's what you think contributes to fibronectin?
That's where we started. I think the other cell types, I mean, we're now doing single cell studies, and so we haven't looked at other cells yet, but that's in the process.
And vascular smooth muscle cells as well.
Yeah.
Astrocytes and smooth muscle cells.
But that was known. It was also known that it was in the vascular.
But for the microvessels, there's no smooth muscle, so probably the astrocytes are the major cell type that affect.
Great, thank you.
All right, thank you very much. Thank you, Richard. Our final speaker before lunch is Dr. Haines from Case Western University. We'll also talk about protective factors.
Well, thank you, and I appreciate the opportunity to come and talk today, and I particularly appreciate being the last person standing between you and lunch. So hopefully we can get through this fairly quickly. One of the things I want to talk about was the potential of an interaction between haptoglobin and APOE in Alzheimer's disease, and we all know why APOE is important. That's why we're here, right? So the question really is, why look at haptoglobin at all? And so there's a series of ideas that we sat down and talked through with this based on some observations that I'll talk about in a little bit. And one is that free hemoglobin, is prone to autooxidation and is a potent pro-oxidant. It scavenges hemoglobin and thus reduces oxidative stress. Oxidative stress is important. APOE lipid clearance is influenced by oxidative stress. Haptoglobin and APOE proteins can physically bind to each other. It's been shown in CSF. And haptoglobin influences the interaction of APOE and A beta. That's been shown in autopsy brains. And haptoglobin can alter A beta uptake. That's been shown in astrocytes. So there's a biological rationale for thinking that maybe haptoglobin might have something to do with anything. And then there was a set of observations that were made not in Alzheimer's disease, but in fact in individuals that are HIV positive, and where they looked at haptoglobin alleles and saw that there was an impact on the neurocognitive impairment in those individuals. And through that and through working through what could be done with haptoglobin there, we thought, well, why not take a look at Alzheimer's disease as well? And so this is based on some work that was done by Haines by looking initially at the HIV positive stuff and used in Will Bush's lab. So a lot of this work is coming from their lab. The haptoglobin gene, there are two major variants of the haptoglobin gene, haptoglobin 1, haptoglobin 2. The main difference is the duplication of exons 3 and 4 in the haptoglobin 2. So we've got a bit of a duplication effort going on here. And that makes it a little bit more difficult to actually haplotype and to actually measure and genotype. So there's a common CNV that spans the two tandem exons. It distinguishes structural alleles. There's that haptoglobin 1 and haptoglobin 2, which has two copies of exon 3 and 4. The difference in the two alleles leads to different protein conformations. So if you're a haptoglobin 1-1, you can make it essentially a dimer, and that's pretty much all you get. Once you introduce the haptoglobin 2 allele, then you can get all sorts of different conformations of haptoglobin, which again suggests that there may be some functional differences between the two. So this again leads us to think that maybe there's some functional reason why we would want to look at haptoglobin. So, there have been a lot of genome-wide association studies that have been done, a huge number of those that have been published, and haptoglobin hasn't shown up in any of those. And part of the reason for that is that haptoglobin is hard to genotype, and partly because it's a duplicated region. So it doesn't genotype well on the SNP arrays because of that structure. So one of the things I did was go in and look at all the GWAS to see whether or not there was any other SNP that was in that same region, just outside the gene, that might have shown an association. You look at all of that, and there's basically nothing there. I mean, the most significant nearby association is modest at best, doesn't make genome-wide significance, and is a little bit downstream of haptoglobin itself. So then you say, okay, those are the SNP arrays. What about whole genome sequencing? The problem with short-read whole genome sequencing is this is a duplicated region, and so it's hard to actually pull it out of the whole genome sequencing as well. So again, it's been a bit hard to look at. So one of the things that in particular that Haines did was to figure out how you could impute the HP alleles using a custom set of reference panels. And I won't go into the details of that. And so we could impute that using the top med and then doing hard calls on the haptoglobin 1, haptoglobin 2, using a dosage greater than 90.9. So pretty stringent criterion. And this was also validated in some samples using the unique exon junction in the RNA in some RNA-seq data. So the imputation seems to work really pretty well and can be done on the SNP arrays and now also we think on the whole genome sequencing. So what did we actually do? We analyzed the ADG data set. Thank you, Jerry. And you'll see here that we have approximately 20,000 individuals that we looked at, and almost 10,000 cases, 10,000 controls. So this is the basic ADGC data set. The genotype frequencies here, just to give you an idea, I've got it broken down by the combined genotypes, but the most important, I think, is the summary of the haptoglobin genotypes, the 1-1, the 2-1, and the 2-2. And those frequencies are pretty much what one would expect out of a European sample. We'll talk a little bit about other samples in a minute. But so we're getting we're getting pretty much what we would expect for the allele frequencies. But now we have to figure out how we're going to look at this, because we don't think that there's a direct relationship, a direct single main effect of haptoglobin and Alzheimer's disease. So we sat down and spent a lot of time and a lot of conversations about how we could possibly look for this interaction. As most of you who do a lot of genetic analysis know, looking at gene-gene interaction is not an easy thing. It's very hard to figure out how you can actually try to show this. So what we decided to do was to generate actually a new variable to look at the APOE 2, 3, and 4 alleles, and basically the linear relationship of this, essentially assuming that the 2 is protective, the 3 is neutral, and the 4 is risk, and just making that a linear thing. Now, we know it's not exactly linear. We did actually look to see whether that made a big difference. It doesn't seem to, so we just went with the linear relationship. So we did two basic analyses on these data. The analysis one was basically just a test for the haptoglobin, the main effect of haptoglobin frequent. Haptoglobin's got age, sex, and 3 PCs in there. The analysis two is, let's see if we can see any evidence of the interactions. And to do that, we threw everything in there. So we've got haptoglobin, we've got the linear relationship for allele 1 and allele 2, APOE 1 being allele 1, which is your first allele for the E2, 3, 4 effect. Age, sex, the PCs, and then all the possible interactions that could be there. And analyze that in the ADG data set to see whether or not we could see an effect. And so what we see in analysis one, pretty much what we expected was there seems to be no main effect. When we look at analysis two, which is the one that includes all those interactions, we see what we expect to see, which is that the effect of APOE, the APOE alleles, is in fact highly significant. Age is highly significant. And then we see the impact of the interaction of haptoglobin, the haptoglobin 2, we called it the haptoglobin 2 allele in this case, and the APOE alleles. And there's a modest significance to that interaction. So that's what we were really, really glad to see, was that there did seem to be some evidence of interaction. Now, what was perhaps more interesting was that the interaction, and this is in individuals of European descent, seems to attenuate the effect of the APOE2 and the APOE4 alleles. So here you can see that the haptoglobin, the 1-1s and the 1-2s, and then the 2-2s, you can see that the risk seems to increase somewhat with the HP22s, even for in the HP22s when you have APOE22. So it's increasing that risk. Same thing with 2-3, the risk seems to increase somewhat. So we're attenuating the effect of the 2, which is, we all look at as being protected. And yet we get the opposite effect when we have the 4 allele. So it's attenuating the effect of the 4 allele as well. So it's bringing both of those back toward neutrality a little bit, which we found, really, really quite interesting. If one takes a look at the other APOE genotypes, there seems to be no effect on the APOE 3/3s, virtually no effect on the 3/4s, and maybe a slight increase in risk with the 4/4s. So not quite sure what's going on with that, but it seems to be that the main effect here is on individuals who have the APOE2 allele, that haptoglobin seems to attenuate the effect of the other allele, and then you can see all 6 of those. Now, that was in the ADGC, in the European samples. There was a limited amount of African American samples as well. I'm not showing you the individual level, the individual results for that. But when we did a meta-analysis with the African American samples and the European samples, we see essentially the same trends here. So they're going in the same direction. So it looks like we, initially may have the same kind of effect going on, not just in the European samples, but also in the Alzheimer's samples, in the African American samples. So that's pretty much where we are. We really need to take a look at this in other samples. We need to take a closer look at what's going on with the haptoglobin, the gene itself, and would really like to get some more detailed information about that. But basically what we can say is that haptoglobin and APOE interact biologically, that we know. Haptoglobin by itself doesn't seem to have a strong influence on AD risk. It appears to statistically interact with APOE to alter AD risk. And so we have some biological interactions, we have some statistical interactions, and the haptoglobin 2 allele appears to attenuate the impact of both the E2, i.e. making it less protective, and the E4, making it less risk, at least on the APOE2 background. And so with that, I think I would just like to thank everyone who worked on this project, particularly the folks in Will Bush's lab. And I'm happy to take any questions, although I won't be offended if everyone wants to go to lunch. All right, thank you very much.
Thank you. We have time for any questions? Any online?
No, but I'm going to ask one.
Okay. And you have your microphone on.
I do, yeah, I remembered this time. So very interesting, Jonathan. I wonder whether you are able to look at any biomarker data connected to people in ADGC or other data sets to see whether that interaction that you're seeing on risk is having some impact on AD biomarkers.
Yeah, that's a wonderful idea. We are actually trying to pull together that data now, and we're actually trying to look at a couple other data sets where some of that data exists so that we can actually take a look at this. But one of the things we have to do is we have to go back and we have to call the haptoglobin alleles and all of those other data sets before we can do the analysis. But yeah, that's a great idea.
All right. Well, we're ahead of schedule, amazingly. So thank you for all the speakers this morning. I guess we're breaking for lunch. We'll be back we're scheduled to come back at 1:15. We'll have 2 further genetic studies, and then we'll move into mechanism of disease. So thank you very much. We'll resume at 1:15.
these two APOE genotype groups. And E2 carriers is in GWAS, one of the gene is near genome-wide significant, and especially this risk allele A is increasing expression of the particular gene PPP2CB in whole blood. This PPP2CB encoding C subunit, which is a catalytic subunit of PP2A enzyme. This enzyme is complex and actually the phosphorylate tau, which is very relevant to the Alzheimer's disease. And in brain tissue, PPP2CB protein level is strongly correlated and associated with C4b, which is complement 4b protein. It is consistent all and E23 and 3,4. And a beta 42 level is associated with the PPP2CB is associated a beta 42 level only among 3,4 carriers. And this is also confirmed. The PPP2CB level and p-tau is positively correlated, as well as C4b A and B level is positively correlated in iPSC-derived neuron and astrocyte. What about the transcriptome analysis in E2 carriers? So E2 carriers differential expression analysis identified complement for A and B is most significantly different among E2 carriers. So it seems this differences in AD is increased and control is downregulated among E2 carriers is only among E2-3. You did not see 3,3,3,4 these differences. This means this protective effect of E2 is somehow involved in the increased level of complement 4A level. And the protein level is also the p-tau protein level is associated with the complement 4A, 4B. But it's interestingly, the expression level is not strongly correlated with the protein level. And also the gene co-expressed genes among these E2 carriers is one particular network was only one significantly associated with the AD versus control in this group. And we looked at the network of genes in this particular gene expression network is co-expressed together with the CR1 and HBEGF. This means E2-related co-expressed genes is actually indeed connected to CR1, and then the HBEGF is both also related to immune response or complement pathway. Now the E44 carriers in GWAS identified 4 genome-wide significant hits. In particular, the most significant SNP and is protective against the AD is actually increased in expression of TRB2 among E4 carriers. This protection is confirmed in the cognitive scores of these carriers, which is protective against the cognitive impairment. And this is also confirmed in the iPSC-derived neurons and astrocyte is increased level is identified in E2 carriers compared to the E44 carriers. And this is consistent in eye neuron and eye astrocyte. In conclusion, tribe 2 expression is increased and the protective allele is increasing in this tribe 2 expression. And tribe 2 seems related to the T-cell immunity. And what about the epigenetic profiles? And we confirmed actually the E4 carriers versus non-carriers epigenetic profiles most different in the APOE locus. It's not surprising. But surprising thing is, and the CpG site in APOE is hyper, which is a higher methylation compared to the E4 in E4 carriers compared to the non-carriers in both blood and brain. So this is blood and this is brain. And the APOC CpG site, it's significantly different between E4 carriers, non-carriers, but only among the only in blood tissue. On the other hand, TAM40 is hypomethylated only in brain. What it means, we can actually disentangle this loci because previously we cannot disentangle using just genetic variant itself. So in epigenetic profiles can distinguish this effect of these genes in blood and brain. So this is new direction we need to consider. And this co-methylated genes is strongly associated with enriched in the estrogen response and UV response. In summary, it actually protective mechanism related to E2 allele. It seems related to tau phosphorylation or dephosphorylation, as well through the classical complement component, especially C4. But distinct from the E44 carriers protective mechanism, it seems it's a related new protective mechanism and somehow linked to T-cell immune response. And epigenetic profiles regulated in E4 is also very distinct in the methylation profiles in this APOE loci. So thank you very much for my lab members, especially Rebecca and Sammy, to do the old work. Thank you very much. Cannot hear. We cannot hear. You cannot hear. Can you turn on the conference room?
Oh,
Thank you.
Can you hear me now?
Yes.
So
Okay, good.
yeah, so
Yes, sorry I
more
cannot
than-
be there. Yeah.
So I know that Allison, I was involved in a paper that came out of your lab showing about maybe 12 years ago that there was a risk variant in PP2A that modulated levels of both A-beta-42 and tau and CSF, and it was in PP2A. I wonder whether this mechanism might be related to what we found previously with APOE2 now.
Oh, who's speaking? Sorry. It was great. Yeah, I really want to learn more about what you mentioned, and it is really interesting to understand the PP2A has some. I saw some PP2CA because it's the most abundant and a lot more studied than PP2CB. So the expression level differences in PP2CB and CA mostly related to brain samples. So, but we probably need to take a look at closely and what differences in PP2CB and CA. But it is really, I believe it's the same pathway. Probably we are kind of pointing out. Yeah.
Yeah, I can send you the paper, Gyungah, when it might be interesting to look at that SNP in conjunction with the SNPs you've looked at to see whether there's any interaction at all or additive effect.
of these variants.
Exactly. Yeah, we saw the brain tissue protein PP2CB level is highly correlated with the PPP2CA, but only difference is it's not APOE genotype dependent in PPP2CA, which we need more larger sample size to disentangle these differences between PPP2CA, CB. What's the role of PPP2CB? Unfortunately, I haven't gone too far from here to molecular findings. Yeah.
Alan.
Data?
No, Alan.
Alan.
Hey. Thanks so much for a great talk, Gyungah. It's an incredible work. Alan Renton, Icahn School of Medicine at Mount Sinai. Should have started with that. So I just wanted to mention, comment on your C4A, was it? C4A or B nomination. So I'm not sure if you're aware, people in our center led by Kurt Farrell and John Crary's lab and various other ADSP people recently about to publish a PSP-GWAS paper in Nature Communications where that was actually identified as a locus associated with PSP. So I think it'd be great if we could like swap notes and just discuss about which variant you particularly found and whether we're talking about the same haplotype. And just, I guess, do you have some thoughts generally about what this means more about understanding how complement cascade might contribute to tauopathy?
So it's a great comment because I looked at very closely HLA locus. C4 haplotype is actually MHC3, which is between the MHC 1 and 2. And then the schizophrenia and other studies identify C4 haplotype is really related neurodevelopment and neurodegeneration. And so I believe, just like you commented, that it's very, very interesting, which is right under the HLA locus. So I believe something going on together with the MHC2 there. And then we haven't actually fully understand HLA locus, and which is a C4 is also related, the copy number variant in there. So it's really something I'm actually really focusing on HLA locus and including C4 haplotype, which is a full description of HLA, which we previously, or in genetic field, we probably missed the importance of that MHC3, which is actually C4. Yeah, I love to discuss more about what your thought and your findings. Yes.
Yeah, that sounds great. Sorry, just a related question. So that's exactly what Kurt found actually, was that there was some connection between C4, I think it was A copy number, and PSP status. So have you actually been investigating C4 copy number in this context in your analysis?
Yes, I actually, I'm working on, and then one of my students and Sammy, which she did the E44 GWAS as well. She's actually, her dissertation is related to C4. And we are going to look at all the phenotypes as well as some haplotyping and the HLA locus, including C4 haplotyping, which is the reference is available, through the Nature paper. So we already got developed this pipeline, and then we are investigating. But I haven't actually extended to other disease models, which is very interesting. You pointed it out, yeah. But I was looking at schizophrenia in UK Biobank, but not the PSP. Yeah, it's very interesting. Yeah.
Yeah, let's follow up, and I'll connect you with Kurt, who's leading this paper from Mount Sinai. Thanks so much. Great talk.
Yeah, thank you.
Do we have any online? No. I think, Gyungah, this is Jeff Vance, the emphasis on APOE2 is really needed. We've all agreed on that, but we don't, so I'm glad to see. So you were screening for variants that reduce the protection for APOE2, right? What group did you actually screen? Was that the ADSP data set, or what did you?
ADGC, so the E2 case is very small, so we combined all this is only European, ADGC, so GWAS data. We haven't looked at ADSP, but I'm going to look at ADSP, the whole screening E2 carriers, the whole genome sequencing data, because it initially, this is a part of ADGC GWAS stratified analysis. But that we really need to extend it to, I do agree. Other ancestry is also haven't looked at. Unfortunately, the sample size we are in, even entire world, is very, very low with the E2 carrier state, I mean AD cases. But fortunately, ADGC had some, so we initially look at. But the power is a little bit low. But I believe multi-ancestry data set will help a lot. Yeah.
Yeah. Well, the new set is, I don't know if the new set is coming out, what, in the fall? Is that the latest? So that'll be what, 62,000. So that really up our number. And I think as we increase, as we've heard from different ancestries, that may give us. But I think that the idea of looking at opposite direction on APOE2 is a really great idea.
Yeah. Thank you.
Oh, one more question. Mike's next, but.
Yeah, one of the mics.
Hi, I just had a quick question. Michael Haney from UPenn. You showed the TRIB2 increased expression in the APOE2 carriers. Is that, do you know what cell type expresses this TRIB2? Is it microglia, astrocytes, all the above?
So according to our iPSC cell line, it both expressed in astrocyte and neuron. But I haven't looked at the microglia. I bet that Alison and others has data, but I haven't. Our iPSC lines is derived astrocyte and neuron. Both are expressed, highly expressed. Yeah. But microglia is great to look at because it's a T-cell related, I agree, but I don't have iPS. Yeah, I don't have iPS derived microglia. Yeah.
Great, thank you.
Gyungah, that's great. I think we'll let you get back to sleep. And
thank you very much.
Thank you for joining us.
Yeah.
Our last presenter of the APOE genetics group is Mike Greicius.
Got it. All right. Good afternoon. Thanks, everybody. I'm Mike Greicius. It's goodness gracious if we're doing name pronunciations from Dr. Mayeux. I'm the former, now 55-year-old one-time medical student in Dr. Mayeux's group, and I'll be speaking about rare protective APOE variants today. I want to thank Jeff and Dave and Marilyn for putting this really great seminar together. All right. Right, so a while ago we were asked to do a review of APOE from Neuron. I almost always refuse to do reviews, mostly to spare postdocs because it's a lot of work and it usually doesn't add up too much, but Michael Beloy and my group agreed to take this on, and we sort of framed our take on this review with this question: is APOE4 inherently bad, or is it not just as good as APOE3, which would have obvious implications? And this was sort of generated by this really beautiful case study that came out of UCSF a number of years ago. So this is a person who, as you can see, this is also a nice reminder of the pleiotropic nature of APOE. This person was homozygous for a very rare frameshift stop gain mutation on APOE. They had no measurable APOE in their lipoprotein fraction, so this is really, there's no APOE around. They had heavy duty, as you might expect, cardiovascular disease. These are xanthomas on the patient's hands, just collections of cholesterol. He agreed to undergo a spinal fluid analysis through the UCSF Memory and Aging Center, and he had normal levels of amyloid and tau, but was only age 40 at the time, and in fact had some abnormal memory testing. So to me, this was like a perfectly ambiguous case study. Because on the one hand, the person's very young, there's no abnormal amyloid or tau, they also have some abnormal memory, but it sort of planted this seed that we then pursued, looking for APOE loss of function variants initially across the ADSP. And here we frame this as if E4 is just not working as well as E3, then a loss of function should increase AD risk. So an E3 null should look as bad or worse as an E3, E4. And conversely, if E4 is inherently toxic, a loss of function, particularly on the E4 haplotype, should reduce risk. So in this case, an E3 null should look better than, for example, an E3, E4. And so I'll ask you to try and keep that logic in mind as we go through these individuals. So we looked through 56,000 exomes and genomes available in the ADSP, and here I'll second Richard's comments about how incredibly valuable this whole project has been, I think, to the field and certainly to my group. And of these 56,000 individuals, we only identified 7 loss of function variants, so 6, SNP, loss of function variants, and 1 deletion. And right off the bat, if you just ask, are you more likely to find these in cases than in controls? So we found of the 6 loss of function variants and 1 deletion, 5 of them were in controls. Only 1 was in an AD case, and 1 was in a PSP case. So it's about a 4-fold increased likelihood of finding these, at least in the ADSP, in a control compared to a case. And this is just a table showing the details of these. I'm going to walk through a couple of these anyway, so we don't need to linger on that too long. And then this is just a demonstration of where we found these different loss of function variants. And here it's worth pointing out, so here's this large deletion that takes out exons 1 and 2. The rest are early stop gain mutations. And then there's this interesting one, which maybe we can talk about in the question and answer session, which is a late stop gain at position 228, which we actually excluded. This is marked as a N truncation variant. And we did some cell-based assays to show that it's actually produced and to some degree secreted. So we didn't really include this one in our analyses. Right, so to start with the sole Alzheimer's case that we found, so this person was nominally E3/4, but the deletion was on E3, so we, with some help from collaborators at University of Washington, Cheng and Yu in particular, we were able to phase a couple of these. So this was an E3/E4 nominally, but the deletion was on E3, so this is an E4 null individual, effectively. And again, if E4 risk reflects loss of function, then we expect this E4 null should have an E4 looking sort of phenotype. So their onset was at age 75, and they died at age 87. Their pathology was very consistent with Alzheimer's disease. And just looking at the ADSP ADGC data, the mean age at onset for E4 homozygotes is 68, and for E4 heterozygotes is 75. So this person, obviously can't make too much of this, but was behaving more like an E3/E4 than an E4/4. And our very generous interpretation here is that, knockdown of E3 in the E3/E4 setting seems not to change AD risk much. I think that's about the most we could say from that one case. The rest, I'll keep saying case, that was the only case. The rest are controls, and I have trouble kind of reminding myself of that. So here's healthy control number 2. So they were E3/4 nominally, but with a stop gain on E4. So we call them an effective E3 null. They were cognitively normal at their last assessment at age 79. And I think more importantly, they underwent spinal fluid analysis at 76 and were amyloid negative. And so we asked, how uncommon is that among E3/E4 individuals? And here, this is a really nice study from the group in Amsterdam, looking at about 9,000 individuals who gave spinal fluid. And here, the far left here are healthy controls. This is subjective cognitive impairment, and this is mild cognitive impairment. So among the healthy controls, this is the E3/E4 curve. And by this person's age, they weren't a patient, about 2/3 of people who are E3/E4 should be amyloid positive among healthy controls. So they're in this sort of protected 1/3 that are still amyloid negative. But then we would point out too that this really underestimates the potential protective status of this individual, because there are a lot of E3/E4s that aren't still healthy at this age, right? That already have MCI or even Alzheimer's disease. All right, and then perhaps I think the most compelling of these individuals is healthy control number 1. So again, nominally an E3/E4, but a stop gain variant in phase with the E4 allele. So another effective E3 null individual. They actually came to brain autopsy at age 90. They were last adjudicated as normal at 90, died the same year, came to brain autopsy. And interestingly, they had absolutely no amyloid, not just in the parenchyma as plaque, but also no amyloid in the blood vessels. And so we again asked this question compared to other nominal E3, E4 individuals, how uncommon is this? And here we were able to look at autopsy data from NAC. There were 1,700 individuals in the NAC autopsy or so, in the NAC autopsy database who are E3, E4. So this is all E3, E4 individuals. And you can see from the first plot, in terms of the SERAD score, this person, they're 0, they're the red dot here. They're going to be 0 on all of these. But it's pretty uncommon to find 90-year-old E3, E4s that don't have any amyloid is the bottom line. And this is their solve phase. So this is sort of the distribution of amyloid across the brain. Again, they had no amyloid, so their solve phase is 0, quite unusual. And then lastly, this is their cerebral amyloid angiopathy score. And I mentioned they had no amyloid parenchymal plaques and also had no amyloid in their blood vessels, which a little less uncommon, but still unusual at this age for an E3, E4. So based on these very, I want to reiterate the fact that we're talking about 2 or 3 individuals here, right? There's no statistics. These are all, this is a case series essentially. So based on this, this question of is it a kind of a weak friend or a toxic foe, we're leaning a little bit towards the toxic foe side of things based on these loss of function results. I mentioned these 2 seemingly resistant phenotypes. So nominally E3, E4, knockout of E4 with, amyloid negative phenotypes at ages 76 and 90. I think if nothing else, it seems as though knocking down E4 should be safe. So we found 7 long-lived individuals in the ADSP who, had lifelong APOE loss of function, at least on one allele. But I think, and we'll hear, I think tomorrow about some approaches to knocking down APOE, which I think is definitely worth pursuing. Certainly the animal models, as we've seen, support the notion that APOE might be a toxic gain of function. But obviously it would be cleaner to look at some small molecule approaches to treatment that don't depend on this sort of sledgehammer approach of just knocking down APOE. So we've done this, again, using ADSP data. We were looking at missense variants in ADSP, not obviously looking at APOE2 or APOE4, but these other 3 where we had sufficient, carriers of these to actually to do some statistics. And so of these 3 that we looked at, V236E and R251G, both had a significant protective effect in ADSP and in ADGC, and so we took those on to replication studies. I should mention V236E was reported in 2014 as protective in a study of about, I think, 9,000 individuals from the Mayeux group with Steve Youngkin. So this is referred to as the Jacksonville variant. And we basically replicated this, and I think it was, I can't remember if it was Alison, somebody had said we narrowed the confidence interval here. So they reported an odds ratio of around 0.1 or 0.15. It's probably a little closer to 0.3, but very protective on an E3 background and very consistent across these different cohorts. And then R251G is new. We didn't have the chutzpah, I guess, to call it the Stanford variant, but I think it's more helpful to actually just go with the amino acids anyway. And this is always on, at least in our hands, on an E4 background. So this is an E4 associated allele that's very protective, reducing risk by about 50% on an E4 background, which I think is important to keep in mind. And then, as actually Dave Holtzman had shown the slide too, we kind of compared people with either the Jacksonville variant or R251G against the E33 background, which is set here at 1. Here in red is E23, the protective effect. Here's the Jacksonville variant, even stronger probably than the E2, E3. And then the R251G, again on an E4 background, reducing risk, somewhere between about 50% and maybe 40%. All right, and then I'll just end with this figure and some reminders. So, these 2 variants are both interesting in part because they're on the C-terminal domain, which has gotten a little less press, I think, over the years. And certainly R251G is very squarely in the middle of this lipid binding domain. I think some people here know about this. This is CS6253 is a peptide experimental drug agent that's actually now through phase 1. This is with Artery Therapeutics. And we're particularly interested in it because it's modeled on the C-terminal of APOE. It's an agonist of ABCA1, and it includes this amino acid position 251. So I think this really puts R251G in the spotlight. I'm really glad to hear that Dave Holtzman's group is working on a mouse model. I think that should be really informative. And yeah, I think I'll end there. Just thank my collaborators at Stanford in my lab. Lots of extramural help, particularly on the loss of function paper from Cheng and Yu at UW. Yeah, and then particularly thank Michael Beloy, Jan Le Guin, and Augustine Kemperati, who led most of this work. Thanks. And I don't know if that was 10 minutes or 40 minutes. Sorry if I went on.
You're good. I had a question on your loss of function. So you compared your loss of functions to 3 4s, but there are actually 3s, right? And so if you looked at how they do relative to 3s with the question is they're going to have half as much APOE expressed, presumably. Are they different because of that?
Yeah, so that's a really important point. When we first started writing this paper up, I wanted to call it APOE haploinsufficiency or something like that. And luckily, I'm not a geneticist, if that's not obvious. One of my geneticist colleagues says, you haven't shown that they're haploinsufficient, right? And so none of these patients were at Stanford. We were lucky to get some tissue to do some of the phasing, but we don't actually know if the other allele has ramped up its expression. We have no sense for that because we don't have plasma or sufficient sort of tissue to look at, expression levels and protein levels.
So a related question coming from online, and probably the answer is no. Could there be compensation by other APOE lipoproteins?
Yeah, I mean, I think the answer is yes, but I don't know. Yeah, right. And I don't know if Jeff was angling towards this, but so, I showed you those nice sort of cloud plots of where our E3 null individual lined up against E3, E4s. This is against E3-3, right? It's a little artificial to say we knocked out E4. This person was just born with, we think, one working copy that happened to be E3. But so even when you look at this person as a nominal E3-3, it's a little less impressive, but you can still see that they're,
they're in the lower,
pretty reasonable outlier.
Yeah,
but that's the best we could do.
But it is an important question, I think, because that's, David's brought this up that, 3 is still a risk. And if we drop it, expression
yeah, I
in general
don't think we can, we had the one E4 null, right, who had Alzheimer's disease. So it's a little tough to make too much of that. I'm not sure how to answer that part.
Richard Mayeux, Columbia. Michael, very nice presentation. Are any of the other than
Richard, make sure you, it's on.
Okay. Any of the people, other people with the more protective variants in the APOE, do you have any other data on like autopsy or biomarkers or anything else?
Yeah, so those are much bigger sample sizes, right? In ADSP, I think there was 33 carriers of the R251G, and then more as we expanded. We didn't look, it's something that comes up every 6 months or so. Should we look now? Do you think there's more, biomarker data? I'd kind of eat my hat if it wasn't involved with amyloid processing, but we haven't been able to show that yet. Yeah.
Dado, not Alan from Mount Sinai. It's not statistical, at least conceptually. How do you deal with possibility of selection bias, and especially think about survival effects due to midlife cardiovascular disease? And do you have any data on plasma lipids on these people?
Yeah, so when I saw Dado at the microphone, I was worried it was going to be a statistical question. He said it wasn't going to be, and it still kind of is. But anyway, it's a good question. Yeah, so in ADSP, you can't really address that, I don't think. It's for sure not a population-based study. We looked in UKB only because the reviewers made us. We were very reluctant to do so because the phenotyping is not so hot, as you know, in UKB. And in there, and in fact, I have the slide up, we did find one E3 null person who was a case in UKB. But the problem with UKB, as I mentioned, is that the phenotyping is not great. So this is an early stop-gain mutation. It should be a loss of function. Their diagnosis was unspecified dementia at ages 70 to 79. At age 79, a number of additional diagnoses were added, dementia and Alzheimer's disease, but atypical or mixed type, other Alzheimer's. So this did not sound like a straightforward AD case. I wish we had biomarker data, but it's UKB and we don't. So yeah, I think, UKB gets us closer to, I think, avoiding referral bias. Mayo Clinic study of aging. There are these larger population studies, but we don't have access to those. I'd love to see, what this looks like in that setting.
Did you look at the lipid level data?
Oh, sorry, that was the second part.
It's available in UK Biobank.
Yeah, so we
right?
did look at that in UK Biobank for a couple of these. I mean, even for in UKB, things like R251G, they just aren't that plentiful, or there aren't that many people. I think we might have seen a little bit of an effect. W228 star, this late one that we ignored, is more common. And certainly when people are homozygous for that, they have a dyslipidemia. But heterozygotes don't seem to have much. The other problem with UKB, of course, the National Health Service being an excellent version of socialized medicine in many ways, is that a lot of those people are on statins. And it's hard to, disentangle the statin effects from the genetic effects.
Hi, Eliezer Maslia from NIA. Can you?
Yeah, it's working.
Working. It's very directional. So.
All right, great. I was wondering if you have checked what is the effect on stability of APOE of these variants that you show in the C-terminus, or if you know if there is any less or more cleavage, or does it do anything to protein stability?
Yeah, we haven't looked at protein stability. And when I say we, bear in mind my lab's a dry lab, but I've been working with Chetan Khosla, who's a really world-class protein chemist at Stanford, who it took me a couple of years, but now he's interested in APOE. So he's looking at that, in particular, though, focusing on the interaction between APOE and ABCA1, not so much on stability. So I can't answer your question.
Thank you.
Yeah, Dave.
Yalong Huang from Gladstone USSF. Great talk, Mike. Just one comment following Eliezer's question. So the APOE4 susceptibility to protease cleavage does occur in the C-terminal and around that area. And based on the mutagenesis studies, any arginine mutation in that area will change the cleavage, actually. So this is first comment. And the second, so this R251G mutation in the C-terminal. But as you know, in the C-terminal, there's also weak HSPT binding domain there. So the main HSPT binding domain in the receptor binding area in the N-terminal. But the C-terminal, this area, the arginine change also changed the HSPT binding. I wonder whether it has been tested that way.
Yeah, we haven't looked at, we're just getting underway. I mean, so it's been fun to work with the protein chemists looking newly at APOE, because there's a lot that I had understood about APOE and our understanding of its structure that just wasn't true, for example. So it was nice to see him sort of begin at the beginning. But he hasn't even, approached those sorts of studies yet. I don't know, the heparan sulfate proteoglycan binding, at least that I've looked at it, can kind of cut both ways. So we have a risk increasing variant, R145C, that has sort of the same effect on heparan sulfate proteoglycan binding as the Christchurch variant, but opposite effects on phenotype. So I'm not sure how much to bank on that, but it's definitely something we can look at.
And if I could just, to the questioners, that these mics seem to be very directional, so make sure that you're right in front of it.
Okay, Joachim Herz, UT Southwestern. So I think, Michael, the real take-home message of that very important study you have is that one can reduce the amount of APOE in the brain by 50% without any adverse effects, which is of enormous importance for any siRNA or ASO approaches.
Yeah, thanks, Joachim. I mean, I'd like to make that full-on sort of endorsement, but I'll repeat that we don't actually know that levels are reduced by 2, because we haven't seen if the opposite, the other allele is overexpressing and overproducing. But based on the phenotypes, it feels like something looks quite different. And I do think it should be relatively safe, particularly when we think about the relative lack of safety of other recently approved.
I think, David, in the mice, haven't you seen the same? He nodded.
Great, thanks.
Amazingly, on time. And so Allison and I will step aside here and allow our next moderators to come up.
All right, we'll move to the next session. The next session is for mechanism of disease mediated by APOE. I'm Takahisa Kanekiyo from Mayo Clinic, Jacksonville. I serve as a moderator, and then Dr. Haines.
Hi, and I'm Hien Zhao from Ionis.
Right, and then move to the first speaker. The first speaker is Dr. Dieffenstein from the MIT.
Thank you. I'm gonna switch gear, transition from genetics to gene expression. So we have been collaborating with Manolis Kellis at MIT and David Bennett at Rush Medical Center to do large-scale single-cell genomics analysis in postmortem human brain samples. And one of the projects that we conducted is APOE genotype dependent single-cell transcriptomic analysis. So in this particular study, we obtained postmortem prefrontal cortical samples from the RAS-MAP cohorts, including 12 APOE33, 12 APOE34, and 8 APOE44 subjects. So from the samples, we annotated a total of about 160,000 single-cell transcriptomes. So, so I'm going to show you a couple pieces of data. So obviously, we perform differential gene expression, and these are organized by gene ontology, biological processes, and looking at how APOE4 affects gene expression compared to APOE3. So looking at the 6 major cell types in the brain, we clearly see cell type specific upregulation and downregulation of various biological processes. And in addition, we also observed global upregulation caused by APOE4 genotype in all of these different cell types. These upregulated processes include pathways such as amyloid beta formation, I-kappa-B, and F-kappa-B signaling, and so on. So it turned out that many of these processes affected by APOE4 genotype, they actually already been previously established to be associated with APOE biology. So such as amyloid beta clearance, amyloid beta formation, cholesterol efflux, cholesterol transport, and so on. So knowing that APOE is an apolipoprotein and it binds lipid and transport lipids, so we perform a lipid and cholesterol processes specific analysis. So here it is very clear that you can see among the different cholesterol and lipid metabolic processes, a very clear cell type specific upregulation and downregulation of these processes. And I want to particularly point out that cholesterol biosynthesis pathways are very profoundly upregulated in APOE4 oligodendrocytes. But interestingly, this very same pathways actually show a trend of downregulation in the APOE4 astrocytes. And looking at the cholesterol gene activity, there is a striking APOE4 dose-dependent effect. So here, if you look at APOE33, 34, and 44 genotypes, you really see a gradual rightward shift of this cholesterol gene activity. So with this result, we obviously look to see whether there's difference in the cholesterol abundance in oligodendrocytes carrying APOE4 genotype. So I'm showing you one piece of data where we use the isogenic APOE3 and 4 isogenic IPS lines derived oligodendroglia, and then using for DP cholesterol to label cholesterol. Here you can see there is a tremendous increase in this DP cholesterol signal in the APOE4 oligodendroglia. So another thing is that this increased cholesterol gene activity shows the inverse relationship with the expression of genes involved in axonal myelination. So with this data, we look to see whether there is any evidence of myelination defects in APOE4 carriers. So, here we are using transmission electron microscopy to look at postmortem corpus callosum samples from E4 versus non-E4 carriers. And I think you can appreciate there is this disruption of myelination and thinning of this myelin sheath in the APOE4 corpus callosum axonal bundles. And the G ratio is very significantly increased in the APOE4 carriers. So the obvious question is whether this increased cholesterol activity and accumulation in oligodendrocytes is causative to myelination defects. So, so we identify a small molecule known as 2-hydroxypropyl beta cyclodextrin, which is a drug that's known to promote cholesterol efflux from the cells. And with cyclodextrin treatment, we found that in the E4 oligodendroglia, cholesterol accumulation is very much reduced. So in the in vitro reconstitution system by co-culturing oligodendrocytes and neurons, we were able to show that this treatment actually can restore axonal myelination. But this is also true in vivo. So using the APOE4 humanized APOE4 knocking mice, we first show that these animals, like the human E4 carriers, they have axonal myelination abnormalities. But administration of cyclodextrin for several weeks, this treatment restores myelination in the APOE4 knocking mice. So here you can see the G ratio is significantly decreased in the cyclodextrin treated group. So I think this result really strongly suggests that cholesterol accumulation in the oligodendroglia contributes to myelination defects, and we can use any approach to reduce cholesterol accumulation. This can ameliorate this myelination defect. So, so I also want to make a similar point, but now in a different glial cell type, microglia. So, so we also use the E3, E4 isogenic IPS cells to induce microglial-like cells. And here you can appreciate that in the APOE4 microglia, they show increased lipid burden as evidenced by DP staining compared to the APOE3 microglia. And by the way, this microglia also show functional deficits in sensing neuronal activity and neuronal surveillance function. And Michael Henley also published a very nice paper showing that this APOE4 microglia actually increases tau hyperphosphorylation in neurons. And obviously, looking at the gene expression of this APOE4 microglia, you can see upregulation of various genes involving lipogenesis, such as ACSL1. So, so we use the ACSL1 inhibitor to treat this APOE4 microglia, and this can very markedly reduce lipid
accumulation. And what we found that this treatment actually restore APOE4 microglia neuronal surveillance function. So again, indicating that lipid dysregulation has functional consequences in the normal function of the glial cell type. So, so in the last minute of my talk, I'm going to make one last point. So, so this is data from our most recent effort in using single-cell transcriptomic approach to interrogate a large number of postmortem human brain samples, in this case 427 of them, prefrontal cortical samples from the RAS-MAP cohort. And you can see that the samples really cover a wide spectrum of various Alzheimer's pathology and cognitive impairment. So, so when we compare gene expression differences between brain samples with high AD pathology versus low AD pathology out of 2.3 million single-cell transcriptomes, we identify 5 different biological processes that are significantly perturbed in Alzheimer's disease. And one of them is lipid metabolism, okay? And if we just provide a little more granularity here, looking at regulation of cholesterol biosynthesis pathway, as well as lipid metabolism among various neuronal subtypes, as well as glial cells, you can see this very striking downregulation of cholesterol biosynthesis pathways in astrocytes in Alzheimer's disease. There's very strong downregulation. And on the other hand, if you look at oligodendrocytes now, again, you see this trend of increased expression of this cholesterol biosynthesis gene. So this result really mirror what I just showed you in the APOE4 carriers, where we see upregulation of this cholesterol genes in oligodendrocytes and downregulation in astrocytes. So, so in summary, I show you that APOE4 is associated with cell type specific perturbation of lipid cholesterol homeostasis, and oligodendrocytes show global upregulation of cholesterol biosynthetic pathway, which is associated with reduced myelination and restoring lipid homeostasis by promoting cholesterol efflux, ameliorates myelination defects in oligodendrocytes. And similarly, I show you restoring lipid homeostasis in microglia. In APOE4 microglia can also restore microglia neuronal surveillance function. So I think that this results indicate that lots of lipid cholesterol homeostasis contributes to the pathophysiology of APOE4, mediating neurodegeneration. And finally, our large single-cell transcriptomic data set also reveals similar lipid dysregulation in Alzheimer's subjects, suggesting that loss of lipid homeostasis constitutes an important pathogenic step in Alzheimer's disease. So with that, I would like to thank all the people contributing to the work, my collaborators, and various very talented former and current trainees in the laboratory. And I would also like to thank the funding agencies for supporting our work. Thank you.
Thank you for the great talk. Are there any questions? Gil
Diparola from Denali. Beautiful work, Li-Hui. Question about the potential cell autonomous role of APOE in oligodendrocytes, particularly in relation to the myelin defects that you've seen in the E4 carriers. Do you think that the myelination defects are essentially the result of cell autonomous expression of APOE in oligodendrocytes? Okay, can you also, do you have a model whereby essentially microglia with E4 expression can sort of participate in the demise of myelin in the E4 carrier?
Yeah, definitely. Microglia clearly play a very important role. I think that even in our IPS work, right? While we have APOE4 oligodendrocytes, but the so-called cell autonomous, if you mean that it's intracellular APOE or secreted APOE, you know, we cannot rule out it's secreted APOE, and it can still bind to the APOE receptors expressed by this oligodendrocytes. So, so I personally believe that APOE mainly functions through binding to lipid cholesterol. It's a secreted protein. So, so I think even in a monoculture, I think it still functions in this fashion.
Great, thank you.
Hello. Hello, thank you for presentation. Sebastian Malaves from NextCure. I just trying to understand APOEs when involved in plaques, it's unlipidated. And here you're showing that when there's APOE, there's more cholesterol in those microglial cells. So why APOE is not picking up on those cholesterol and get lipidated? Just like the more lipid you have, the less lipidated APOE you have.
I didn't get it. Can you repeat the question?
Sure. The plaques contain non-lipidated APOE. The macroglial cells here in short produce more cholesterol in presence of APOE, yet you have less lipidated APOE. So it seems like the more cholesterol you have, the less lipidated APOE you get.
Oh, I see. So your question is why APOE4 oligodendrocytes have increased cholesterol, and yet there is less lipidated APOE in this APOE4. So, so APOE4 is known to have reduced lipid binding property, right? So, so I think whether there's intracellularly there is more cholesterol or not. In fact, I will argue because of the reduced lipid binding property, it actually results in you know, abnormal regulation of cholesterol biosynthesis and leading to increased intracellular accumulation of cholesterol, resulting in less transport and efflux.
Thank you.
No question online, but actually I have a question. So did you look at the gender effect on the cholesterol, you know, biosynthesis endpoint? Like,
have I looked at
like, is there a difference in male versus female? Because it looked like in your population you had kind of half male, half female. So is it a general defect that you see regardless of gender? Or...
Yeah. So from our APOE-dependent transcriptomic analysis, you know, we only look at 32 individuals, so so we simply don't have enough power to look at a potential gender effect. But with now the larger data sets and also other people's data set, I think there should be another visit to look at potential gender specific effects.
So I have one question. So you say the astrocytes, the cholesterol synthesis is suppressed, the oligodendrocytes are increased. So do you think which is first? Astrocyte disturbance is first, or oligodendrocytes overexpression first? Do you think as the order of the progress of the disease?
Oh, so what is the order? So whether that's a very good question. I don't really have enough data to make any prediction. I mean, astrocyte is known to be the major cell type producing APOE. Oligodendrocytes, you know, a subpopulation of oligodendrocytes express APOE as well. But the sequence of events, I will have to think that astrocyte probably plays a major role in driving a lot of this different things. But I don't have evidence to say that.
Thank you. So we need to move to next. So the next speaker is Dr. Alison Goate. Oh, sorry. The next speaker is Dr. Alison Goate from Mount Sinai.
It changed the font in the slides. Yeah, it's too large.
Maybe it's only that slide.
Yeah. I hope.
Yeah, me too. All right. Thanks very much. So, yeah, I'm going to talk about the impact of APOE genotype on microglial APOE expression or microglial gene expression and function. And hopefully that will be the only slide where the font was changed. So several years ago, when Julia TCW was in the lab, we published a paper looking both at population level IPS lines and isogenic lines, looking at the impact of APOE genotype on gene expression. And one of the things that we noted in that paper but didn't really delve into was that we could see some background effects that were on gene expression that didn't appear to be due to the APOE protein isoform effects. And so after Julia left the lab, Dr. Anna Podlesny-Drobiniok continued some of these analyses, and she used a mixture of population lines and isogenic lines, and which allowed us to compare the differential gene expression when the APOE isoform was on different APOE backgrounds. So when you put 3-3 onto a 4-4 background, or put 4-4 onto a 3-3 background, or held the isoform constant and looked at the background effects. And what this slide shows is that this is all in iPS-derived microglia. So no disclaimers. I have no idea what happens in any other cell type, but in the context of microglia, we found that the differential gene expression when you were comparing 4-4 versus 3-3 was different based on the APOE background. So if it was on a 3-3 or 4-4 background, whereas when you compared 3-3 and 4-4 and you held the background constant, you actually saw the same differential gene expression for 3-3 and 4-4 on the background. And so that really suggests, at least in microglia, that there are some differences probably in function that are related to the background in addition to the protein isoform change. And I think we don't know about this in other cells, but I think it's something for people to be mindful of, particularly if you're using isogenic lines, because obviously you're putting the isoform on a different background. And we know that the haplotypes that 3-3 or APOE3 are on are totally different from the ones that 4 is on. So in a natural situation, nobody has a 4 on a 3 haplotype background. So then so that's obviously just showing you a very visual picture of how things change overall. But this shows you the specific pathways that are affected. And what we see is that IM jails with an E4 SNP on an E4 background are somewhat different from those you see with an E4 SNP on an E3 background. And what we were particularly interested in is obviously is the differential expression on the E4 background, where you can clearly see these upregulation or positive enrichment for lipid droplet organization and negative enrichment for lysosome, cholesterol efflux, and translation initiation. So the next experiments that I'm going to show you are all looking at population lines comparing 3-3 to 4-4 and looking at functional outputs from these pathway-based analyses. So here this is looking at myelin phagocytosis, and you can see here, over here, this is the readout from Incocyte showing the kinetics. Blue is the E3 and 3-3, and red is the 4-4. So overall, they're not statistically significant based on 5 lines for the 3-3 and 4 for the 4-4. But it does look somewhat lower, but not significant with the number of lines that we have there. So when we look at lysosomal mass, we can see in the middle here, using LysoTracker, that there's a substantially lower lysosomal mass associated with the 4-4 lines compared to the 3-3 lines, but doesn't really seem to be any difference in the acidification or proteolysis in these experiments. So specific effects on lysosomal mass, which might affect both lipid and protein degradation in E4 lines. When we compare the levels of expression, you can see that in these microglia that E4 is expressed at lower levels than in E3 pretty consistently across these lines. If we look at the lipid droplet accumulation, you can see in the population line, we really don't see a difference between the accumulation of Modifier in the 3-3 versus the 4-4, which you can see in the isogenic lines that they're shown there, that there does seem to be an increase from 3-3 to 4-4. So this seems to be like a protein isoform a specific effect as opposed to the background effect that happens on a 3-3 background. So the last phenotype that we looked at in these lines was protein translation, which was also downregulated. You can see the click chemistry gel on the left here, and on the right are some of the plots. And so here you can see that at baseline, so the translation levels in E4, which is this one here, are lower than the translation levels in the 3-3. When you treat with sastagargan to induce the stress response, you can see that they're normalized. We don't see a difference in their response under that circumstance. But at baseline, there are different protein translation rates between E3 and E4. So in the last couple of slides, I just want to move on to some single cell data derived from from isogenic lines. So now looking at the impact of the protein isoform, and yes, so that 4-4 should be over here. This is on the left. We have, so these are microglia and iPS-derived microglia there, and then single cell RNA-seq. And on the left, you see the data from looking at the different microglial subclusters in 3-3, and on the right is the 4-4. And I think you could say the cluster that is DAM is this cluster here. And even if you downsample to have the same number of cells in the two, you see that there's a decrease in the number of cells in the E4-4 that are in the DAM state. And that on the right here, this is comparing the major genes that are up and down regulated in DAM in these different studies that have been done, and comparing it with what we're seeing in the E4 and E3 here. And you can see that universally the comparison of the DAM between 3-3 and 4-4, even though there are smaller numbers, you also see that there's also lower expression of the genes associated with the DAM signal within these cells in E4. So that's a double whammy there, that there are fewer DAM and also lower expression of the DAM genes within those cells. Lastly, this in this experiment, Anna so we knew that these processes were largely negatively enriched in the E4/4 compared to E3, and then we tried to rescue these by treating them with LXR agonists, and I think you can see that the gene expression in the 3/3 and the 4/4 now look very similar to one another after treating with the LXR agonist, and this resulted in a you can see that lysosomal mass was increased and acidification. And so just to summarize then, E4 isoform and the haplotype background influence gene expression in microglia. The pathways that are differentially regulated between E4 and E3 include lysosome, cholesterol efflux, lipid droplet organization, and translation. And in E4 in microglia is associated with lower expression, reduced lysosomal mass, and lower protein translation, which is consistent with the observations we made in the pathway-based analysis. So there are lower frequency of DAM microglia in these in the E4 compared to E3, and that the LXR agonist treatment appears to rescue those gene expression changes that are different between 3/3 and 4/4 in microglia. And lastly, I just want to recognize, I mentioned Anna Podlesny-Drobeniok. She carried out a lot of this work. The protein translation analysis was done by Francesca Goretti, and the original work that started off all of this was carried out by Julia TCW, and she's in the lab. Thank you.
Thank you, Alison. I just want to clarify. So did you use the IPS line from a healthy individual or from AD case?
They're a mixture.
Mixture.
Yeah.
So
and I don't know whether we've, in these analyses, I don't think we've separated out. It was used as a covariant in the analysis, but I don't know that we looked directly to see whether there was a difference. I mean, obviously with IPS, we kind of assume that you removed the epigenetic differences when you made the lines, and so that probably wouldn't be important, but I don't know.
I just wonder that when you compare the isogenic line with the E3 background, the E4 background, if you analyze only AD cases, or if you analyze only healthy individual cases, this difference will be different.
Yeah, I mean, I can't say the impact of AD versus control on any of the things that we looked at.
Hi, Yun from Washington University. So I'll ask a very kind of fundamental question for this pipeline. So how do you know that the APOE isotype will not impact on the differentiation of your iPSC-derived microglia? Like, yes, for this unique pipeline. Because the differentiation from iPSC cells to microglia is different from their original differentiation.
Yeah, no, that's definitely true. We haven't seen any growth effects on differentiation by looking at markers, but it's certainly possible that there is some difference in differentiation. And you're right that the differentiation that we're using here is certainly not exactly what happens to a microglia. First of all, it's in isolation in a dish, right? We don't have the other cells of that communication, and certainly it's possible that these results would look different if you were looking at them in the context of other cells.
Yeah, thank you.
I have a couple of online questions. So first is, could the phenotypic differences be partly explained by the different expression levels of APOE in the 3/3 versus 4/4 line?
I mean, it certainly is possible, right? But I think it's important. I think it's also true that looking in human tissue as well, that we see these differences in expression levels. So I mean, so trying to distinguish between what is due to level of expression versus isoform is difficult. Obviously, in an isogenic one, you might probably be able to isolate that to the isoform effect as opposed to the expression level. But I think it's probably important to think that both of those things are contributing to, right, the expression level and the isoform are likely to be contributing to some of the phenotypes we're looking at.
The second question is, what LXR ligand or agonist did you use?
It's a commercially available one. I don't remember off the top of my head. Yeah, it's got 4 or 5 numbers in it. GO something. I don't remember what it is. Yeah.
And then another
I can look it up and put it online.
Do you find it surprising that you have a reduced lysosomal mass but no effect on phagocytosis? Like it's
well, I guess as I try to point out, I mean, even that you know, they're not statistically significantly different from one another with the numbers we have, but it might be if you had larger numbers, right? I mean, they just that.
Hi, Michael Haney from UPenn. Really great talk. I just had a quick question. It looked like most of the studies on the iPSCs were done kind of like a resting steady state. Have you had any challenges to these lines, like LPS or amyloid, to see if there's differences in the phenotypes, either in the population or in the isogenic lines? I'm wondering in the population, there might be like expression changes in the background that could be read out there with challenges that would more normalize.
Yeah. So the only challenges we've done really are with myelin. We haven't looked at LPS or A beta in these models. I couldn't tell you, unfortunately, what happens in that situation.
Thank you.
Thank you, Alison. So we need to move to next. Okay, so next speaker is Dr. Julia TCW from the Boston University.
Yeah, and our finding is continuing influencing in the disease status. That's how I view it.
Another, oh.
Okay, so we close.
Thank you.
Julia. So next speaker is Dr. Chris Graff from UCSD. I think this is a pre-recorded session.
I'd like to thank the organizers for the opportunity to participate in this meeting and to present some of our recent work. I'm sorry that I can't join you today, but I look forward to joining you tomorrow.
I guess I'll make the announcement after.
Genome-wide association studies have consistently found that most of the common risk alleles for Alzheimer's disease reside in non-coding regions of the genome. This creates challenges for determining the cell types in which they act and the genes that they affect. To address this problem, we and other laboratories have worked to determine the extent to which non-coding risk alleles intersect with transcriptional promoters and enhancers. As one example of this, Alexi Knott, Inga Holtman, and Nicole Kufal led an effort in my laboratory to map the active enhancers and promoters in neurons, astrocytes, oligodendrocytes, and microglia in the human brain by combining fluorescence-activated nuclear sorting with epigenetic assays that are associated with open and active chromatin. Using these methods, they were able to identify tens of thousands of putative enhancers that were specific to each cell type. By performing linkage disequilibrium score regression analysis, we found that while risk alleles associated with psychiatric traits were most strongly enriched in neuronal enhancers, risk alleles for Alzheimer's disease were most strongly enriched in microglia enhancers. This finding has been made independently by several laboratories and supports the concept that many AD risk alleles exert their phenotypic effects by altering gene expression in microglia. What I would like to discuss in this short talk is an alternative approach to leverage enhancer landscapes as a basis for discovery of mechanisms driving disease-associated microglia phenotypes. This approach is based on the concept that enhancers play critical roles in mediating changes in cell fate or cell state in responses to external signals. Enhancers integrate these signals by providing the sites of action of specific combinations of cell-specific and signal-dependent transcription factors. Changes in cell state or cell fate typically involve changes in the activities of hundreds to thousands of enhancers. We realized several years ago that by defining the set of enhancers that become activated in response to environmental transitions and determining the sequence motifs embedded within these enhancers, we could infer the identities of the transcription factors driving these cell fate or cell state changes. I would like to illustrate this general approach to decode microglia phenotypes by presenting collaborative studies with the Holtzman Laboratory investigating the TE4 mouse model of neurodegeneration. As described by Dave Holtzman, the combination of PS19 tau and human APOE4 in this model drives severe neurodegeneration that is strongly dependent on microglia. Working with Maud Gutos in the Holtzman Laboratory, Johannes Goletsky performed a single nuclear RNA-seq of microglia from the hippocampi of APOE4 and tau APOE4 mice and identified a previously undocumented microglia phenotype that was highly correlated with neurodegeneration, which we called tau APOE responsive microglia, or TERM. These microglia express several hundred genes that are not expressed in microglia that are associated with amyloid plaques, and they also exhibit marked cytoplasmic and lysosomal lipid accumulation. The specific association of terms with the combination of APOE and even tau raises the question of whether terms are anatomically or functionally related to the neurodegeneration observed in the TE4 model. To address this question, Joy Zhao designed a probe set targeting of about 500 genes to interrogate the locations of molecularly distinct microglia phenotypes in the mouse brains derived from E4 only mice, TE4 mice, and APP-PSA1 mice as a model of amyloid pathology using MERFISH technology. Analysis of the gene expression patterns of these probes in these three genotypes allowed clustering of all major brain cell types. Importantly, Joy was able to define specific subsets of microglia exhibiting molecular phenotypes consistent with homeostatic microglia, amyloid responsive microglia, and TERM. The most widely separated phenotypes corresponded to homeostatic and TERM microglia, with TERM exhibiting increased expression of genes including CD9, GPNMB, CLEC7A, and AXL. Joy then mapped the anatomic locations of TERM and homeostatic microglia in each of the three models. Homeostatic microglia are distributed throughout the brains of each model, but are underrepresented in the hippocampal regions of the TE4 brain. TERM are nearly absent from APP-PS1 mice and APOE4 mice, but are abundant in the hippocampus of TE4 mice and reside in close proximity to areas of neurodegeneration. A similar profile is exhibited by GPNMB expression. These findings are consistent with TERM driving and or responding to neurodegeneration in the TE4 model. Based on these findings, Johannes Goletsky sorted Pu1 positive nuclei from the hippocampus of E4 mice and TE4 mice, for which the spatial transcriptomic experiment I just showed you primarily defines as homeostatic or TERM, respectively. He then carried out a TAC-seq analysis to quantitatively identify open chromatin in each subset. From these experiments, he was able to identify more than 900 regions of the genome exhibiting more than a twofold increase in the TAC signal at a false discovery rate of less than 0.05. Regions of the genome exhibiting these features are exemplified in the vicinity of the LPL, SPP1, and BHLHE40 genes, which are strongly upregulated in term. De novo motif enrichment analysis of these genomic regions recovered the DNA recognition sequences for CBP, P1, AP1ATF, MITF basic HLH, and EGR transcription factors. From these studies, we can infer that environmental signals in the context of the interaction between PS19 tau and APOE4 lead to alterations in the enhancer landscapes of microglia that drive the term phenotype. From the analysis of the sequences of the genome that exhibit increased open chromatin, we observe a combination of motifs that represent DNA recognition elements for microglia lineage determining factors CBP and P1, and members of the AP1ATF, TFE, MITF, and EGR families of signal-dependent transcription factors. We are currently validating functional roles of these factors in driving specific aspects of the transcriptional responses of microglia to APP, PS1, and TU4 genotypes. In summary, quantitative analysis of dynamic changes in enhancer landscapes enables the inference of transcriptional drivers of microglia phenotypes. Acquisition of the term phenotype is likely driven by combinatorial interactions between P1-CBP, AP1-ATF, MITF, TFE, and EGR transcription factors, although other factors likely contribute to this transition. Application of this approach to microglia in amyloid pathology and lysosomal storage diseases yields overlapping and distinct motif combinations. Amyloid responsive enhancers in explanted human microglia, interesting, are enriched for AD risk alleles. In principle, this approach can be applied to any cell type in the brain and offers a powerful approach to identifying underlying transcriptional mechanisms. With this, I'd like to thank you for your attention. I have mentioned the major people involved in these studies, and in particular, Johannes Galetsky, Joy Zhao, and Maud Grotius. Thank you for your attention.
Thank you. So Chris, now they can take a. Are there any questions from the floor? I have one question, Chris. So the great talk. So I wonder do you have any idea that if the lipid, because APOE might relate to the lipid metabolism, if the lipid mediated this mechanism?
A very interesting question. We know that lipids are accumulating in the lysosomes of these microglia. And in work that I didn't present, we've also been using this approach to study primary lysosomal storage diseases. And what we've learned from studying the MPS3A mutation, a loss of function of SGSH, we see actually a strong overlap in the transcriptional program and the epigenetic program, which makes us think that lysosomal dysfunction is a driver of the molecular phenotype that we're seeing in the term microglia. And just to give you sort of a sense of the magnitude of that, of the genes that we see dysregulated in the SGSH knockout mouse, which by 8 months of age number about 1,000 genes, there's about a 30% overlap with the genes that are upregulated in the terms. And so our thinking is that some of the phenotypes of the term microglia relate in some way to a lysosomal dysfunction, which could very well be driven by the accumulation of lipid that others have commented on.
Thank you.
Hey, Chris. Very nice talk. This is Mia-san from Intramural NIA. So your TERM populations, I think this could be a really basic question, but how much overlap is there between the DAM and the TERM?
Okay, great question. If I'd had more than 10 minutes, I would have told you the answer to that. So when we line up the genes that are dysregulated in the terms and the genes that are dysregulated in the dams, what we observe is a sort of a continuum of genes that are very, very biased to the dams, to genes that are very biased to the terms, and then there's a group of genes that are all going in the same direction in both of those phenotypes. And the breakdown is about a third are biased towards term, a third are common, and a third are biased towards the amyloid response. And each of those subsets of genes have different functional annotations that are associated with them if you carry out sort of a conventional gene ontology analysis. The genes that are most associated with the amyloid response are very highly enriched for terms related to lysosomal function, oxidative phosphorylation, and translation. The genes that are associated, that are upregulated in both phenotypes have a very strong immune flavor to them. So increased in TNF signaling pathways, increases in cytokine production. And the genes that are enriched in the term population are a little bit hard to figure out, actually. There is an immune signature there, but the enrichments for those categories of genes are not nearly as strong as they are in the other two that I mentioned. And we're really in a process of trying to go through those genes one by one to figure out what they are and what they do and how they might relate to the different phenotypes. And I think one of the goals of this overall strategy is to try and identify genes that are giving these microglia their different functions, and that's something that we're I would say at a very early stage of, and just we don't have an answer to which genes are playing protective roles, for example, and which are driving pathogenesis. So work in progress.
What about enhancer differences, like ataxic or?
Yeah. Okay.
histone?
So the overlap of the enhancers look very similar to what I just told you for RNA. So about a third of the enhancers are the open chromatin regions that are responsive to disease. About a third of them are in common, and about a third of them are very much more biased in one of the two phenotypes. And what's interesting is that the motif enrichment analysis of all of these subsets results in a very similar set of motifs. And so what we are thinking is that the microglia are basically pulling from a sort of a common palette of signal-dependent transcription factors, but using them in different combinations to drive both the overlapping molecular phenotype, but also the disease-specific phenotypes. And it's what is pretty clear is that there is no single transcription factor that we can assign to a DAM phenotype or an amyloid phenotype or a term phenotype. For the most part, a very similar set of transcription factors is implicated in driving those two different phenotypes.
I see. Thanks, Chris.
Hi, this is Yun Chen from Washington University. So my question is, at least from the selected markers you show today for the term, including AXL, CLAC7A, GPNB, these kind of the markers, has already been shown that is activated and highly expressed in the original paper from Oleg Blofsky with you together. By that time, you guys used APPS1 mouse model and also the SOD1 model too. You do see this gene upregulation, suggesting that this type of activation happens in those mouse models. I wonder why this case you don't see the term microglia signature in the APPS1. Is that possible that it's due to the disease severity? If you wait longer, the mouse is more aged, can you actually see that term activation?
Yeah, no, this is an important question because it really relates to whether or not these two phenotypes are really just different points on a continuum of severity, which you're suggesting is a possibility, and I agree it's a possibility, or whether these really reflect two different endpoints. And what I can tell you from our application of the MERFISH technology so far is that the term populations are really only seen in the TE4 model. We don't see the term populations in the amyloid model, but there are genes that are upregulated in both of those phenotypes. So as you pointed out, some of these genes like CLEC7A or CD9, they're upregulated in both amyloid and in the TE4 model. And so they don't distinguish between the two phenotypes. With respect to the DAM phenotype, and we're sort of using that definition as sort of laid out in Ido Amit's original paper and also shown in Oleg Botovsky's paper, cells with that phenotype, the amyloid responsive phenotype, we see that phenotype in the APP-PS1 model, but we also see those cells in the TE4 model. So the TE4 model has DAMs and terms, and the amyloid model just has the DAMs. And so, so that still leaves open the question of whether, the progression to terms is just a linear progression along a severity pathway. And we think that's true to a certain extent, but because we also see this collection of genes that is pretty different in terms of their expression values in the amyloid response, and because the terms seem to be localized spatially in an environment that's different from the DAMs, we think that there are aspects of those programs that actually are quite distinct. And one of the things that we're trying to understand now is what the role of TREM2 signaling is. And we think that TREM2 signaling, may be one of the explanations for the divergence of the open chromatin that we see in these two models, and that's something that we're setting out to explore experimentally.
Sure. Yeah. Thank you.
Hi, Richard Mayeux again. In the ADSP data and the ADGC data that we've looked at, only a handful of those genes actually are known to be involved in immune response or inflammation, and the other genes are not. And is the convergence, I'm trying to understand where the convergence, everybody at postmortem has an inflammatory, that there's evidence of activation, but I'm trying to figure out where the convergence is for these two processes. Is it through amyloidosis, or is it through a direct immune response where you don't need amyloidosis to occur first? It can be a secondary thing. Or is it due to the fact that there are other types of pathologies occurring in people with Alzheimer's disease? That may be a too broad a question, but I'm looking for some sense out of it.
I can give you some initial thoughts on that. So one thing that is, I think, pretty clear from our analysis of these two models is that the response to amyloid pathology isn't clearly a response to amyloid. It looks much more like the DAM phenotype is a response to some sort of pathology that has been caused by amyloid pathology, because we see the DAMs in the TE4 model, which do not have amyloid. And in the TE4 model, where do we see the DAMs? Well, we don't see them at the areas of neurodegeneration that the terms inhabit. We see them more diffusely spread throughout the brain. So I think what the DAM phenotype is related to is some sort of some type of tissue injury that is not associated with cell death. And that phenotype can show up in both of these models. and that we really only see the term phenotype in the context of neurodegeneration and cell death. And so that may be, one of the things that distinguish those. Now, having defined these different phenotypes, one of the questions that we've tried to address is whether we see anything that looks like them in the context of human Alzheimer's disease. And what we have done most recently is to try and develop gene signatures that distinguish the TE4, the term phenotype, from the amyloid phenotype, and then map those onto UMAP representations of microglia that came out of a recent paper from Brad Hyman's laboratory, where they performed single nuclear RNA sequencing of cells that were enriched for myeloid cells in patients with AD who were very carefully phenotyped. And what I can tell you is that we can overlap the mouse signature genes for amyloid pathology and the mouse signature genes for TE4 pathology onto disease-associated clusters in that data set. And we can also map the signatures for the lysosomal disorder. And so, to the extent that, that level of analysis is informative, we do think that these mouse models are telling us something about pathological processes in the human brain.
Thank you. So we need to move to next. Thank you, Chris.
Okay, we'll see you tomorrow.
Yeah, as I was trying to say earlier, if we could just please huddle together outside of the conference room during the coffee break, just to take a picture, a group picture. Thank you.
Oh, yeah.
Come in. I'm just answering the question online, so I think I'm good now. Yeah.
Great. Thank you. Thank you to the organizers for giving me this chance to talk. I just opened my lab at UPenn, but what I'll be talking about today is my postdoctoral work in Tony Weiss-Corey's lab on this damaging lipid droplet-associated state in microglia that seems to be influenced by the APOE4 genotype. So what I'll tell you today really started with this chance observation from a fellow postdoc in the Weiss-Corey lab, Julia Marshalinger, who noticed that specifically in the microglia of the aged mouse brain, these large cytosolic lipid stores known as lipid droplets accumulating in the cell type. She went on to have this Nature Neuroscience paper where she described this lipid droplet accumulating microglia state, or LDAM state, as a very pro-inflammatory and dysfunctional state of the aging brain, similar to a lot of reports we've heard earlier today. I was interested in looking at this in the context of Alzheimer's disease, particularly because of comments from Alzheimer's himself, where in his original description of the disease, in addition to his descriptions of plaques and tangles, he noted that the glial cells in particular formed many large, what you call at the time, fat sacs in the glial cells near the plaques. Today, we would probably refer to these fat sacs as lipid droplets. These are these large cytosolic storage vesicles of lipids that bud off from the ERs. These are not the lysosomal accumulations that were particularly heard in the last talk. But these are excess lipids that bud off, and they're mostly filled with triglycerides and steroid esters, as well as other lipids. And of course, the other reason why I want to look at this in the context of AD is because of the APOE risk factor. So to start, we took a kind of bird's eye view of the situation by getting APOE4 brain tissue from individuals with AD, and APOE3 brain tissue from individuals with AD and age-matched matched controls from Banner Health in collaboration with Eric Ryman. And we did a single nuclear RNA-seq on that. And what we noticed in the microglia from these brains was a massive upregulation of this gene ACSL1, particularly in the APOE4 microglia. This was of interest to us because this is a well-known gene or enzyme involved in this lipid droplet formation. It's known that if you inhibit it, it prevents lipid droplet formation. If you overexpress it in certain contexts, it can be causal for lipid droplet formation. And additionally, through genome-wide CRISPR screens in myeloid cell lines, this is one of the top necessary genes in the human genome for the formation of lipid droplets. So not only does it go up in expression, it's functionally necessary for these lipid droplets. This seemed to be also unique to microglia in the sense that these are the cells that express this enzyme the most in the brain, and that they seem to be influenced, this expression is influenced by the APOE4 genotype. We could also see this at the protein level in these microglia, giving us a functional protein marker of this novel microglial state. And kind of in similar to Alzheimer's original observation, these tend to appear near or around the amyloid plaques. So these microglia expressing ACSL1 were most closely associated to the amyloid plaques, perhaps suggesting that the plaques are inducing this expression. To actually visualize lipids themselves, it kind of returns this kind of old-fashioned technique of looking at lipids using oil red O, which we think is somewhat similar to what these original histology stains must have looked like. And in this context, we do see more lipid-containing cells within the AD brain tissue, and this is slightly increased in the APOE4 genotype, again, with these lipid-filled cells appearing mostly near or around the amyloid plaques. Test whether, like, kind of get the causality of here, we used isogenic iPSC lines in collaboration with Yang Huang's lab. We differentiated into microglia, and we saw in very similar ways to what Louis Tsai presented earlier from Matthias Victor's work, that just at this basal state, these APOE4 microglia contain more lipids by staining with Modifier. But if you challenge them with amyloid aggregates preformed, you see a huge upregulation of this lipid accumulation within these microglia, much more in the 4-4 than the 3-3. And this is actually something that's been repeated and seen by a variety of labs. So Goate and Boo's lab's also seen this lipid accumulation in these microglia upon amyloid challenge. And I point to these other papers because it's really important, since we're all using very different iPSC lines, even different differentiation methods, but this finding of increased lipid accumulation within these microglia in response to amyloid challenge in an APOE-dependent manner has been consistently seen across many labs now, just in the past few years. We also see this in primary rat microglia as well, so it's not something specific to the iPSCs or to any lines. And we see this as it happens alongside the upregulation of lipid droplet proteins such as PLIN2 and this ACSO1 enzyme. So challenging the eye microglia with ACSO1 or with amyloid increases ACSO1 expression. Inhibiting ACSO1 reduces these lipid accumulations within these microglia. And this is not unique to amyloid challenge. So Matler and Jones' lab and his microglia showed that if you challenge microglia with LPS, they also massively upregulate this lipid synthesis enzyme. So what we believe is that this is a de novo lipid synthesis event that's happening in the microglia using deuterated glucose and targeted lipidomics in collaboration with Munther-Boer-Millie's lab, that upon amyloid challenge, we see this incorporation of this deuterated glucose into these lipid droplets, and these seem to be mostly triglyceride species, at least in these cell culture models. In collaboration with Chris Glass's group, we characterized the lipid droplet containing microglia from those that didn't have lipid droplets, and we see this NF-kappa B attack-seq signature within these microglia. A lot of pro-inflammatory chemokines are secreted, and inflammatory cytokines are increased in expression, as well as a lot of defects in microglia function like phagocytosis, and we see this lysosomal accumulation phenotype in these cells as well, and consistent with previous reports of lipid accumulating microglia. But to look at the effect that these lipid accumulating microglia have on neurons, we sorted out the lipid droplet high and low microglia and then used the conditioned media from those cells and put those on neurons in collaboration with Hyung Hwang's group. And what we saw there was that the conditioned media from these lipid droplet high microglia actually induced phospho-tau pathology or signatures of phosphorylated tau in these neurons. And this seemed to be APOE dependent. So if you knock out APOE, you no longer get this tau phosphorylation within these neurons. And so to summarize, we believe that amyloid, beta amyloid aggregates or other innate immune triggers induce the increased expression of ACSO1 in microglia. This leads to an APOE-dependent lipid droplet accumulation in our systems. It seems to be mostly triglycerides, but again, this is limited to cell culture. These lipid droplet accumulating microglia are dysfunctional in their normal states, but they also secrete some damaging factors that get transferred to neurons. We actually think that some of these secreted factors might be the lipids themselves that are getting transferred and damaging neurons. And this kind of fits in with some of the more recent protective APOE variants reports, we believe, in the sense that, we're all familiar with this Christchurch variant. We've had lots of amyloid in her brain, but very little tau pathology. We think that it might not matter how much amyloid is in the brain if the APOE-dependent reaction to that amyloid in the microglia does not convert to this kind of pro-inflammatory lipid-associated phenotype that then gets passed on and transferred to the neurons to cause downstream tau pathology and neurodegeneration. We're following up in the lab now on those types of questions with these protective variants, doing co-culture experience or conditioned media experience, where we're tracing the communication between the microglia and the neurons through exosome reporters, and then doing detailed mechanistic studies on the neurons, as well as these CRISPR screens in the neurons. And some preliminary data suggests that what's actually inducing apoptosis in these neurons from the conditioned media are lipids themselves being transferred that accumulate in the lysosome but eventually cause mitochondrial stress. And finally, we're using some in vivo CRISPR screening approaches to look at how different APOE variants influence lipid accumulation within these microglia. And with that, I'd like to acknowledge Tono's Corey lab and all the collaborators that helped with those work, many of which are here in this room, like Hyung Hwang, Chris Glass, Munther-Boer-Millie's lab, Eric Ryman and Banner Health, and of course our funding. Thank you.
Thank you, Michael. Question?
So, as you quickly mentioned, not only Dado Marcora from Mount Sinai. So as you said, not all lipid droplets are made equal. There are different flavors, TG rich versus cholesterol ester rich. Definitely when in vitro, you kind of stimulate TLR, LPS, A-beta, you see these more triglyceride-rich lipid droplets. But is that what you see around plaques in the brain? I think what...
Yeah, same question I asked in the last. Yeah. So what are the lipid species in the brain? We don't know. It's actually quite challenging to do cell type specific lipidomics within the human brain context. There's been a number of papers showing in vitro they seem to be triglyceride rich. Gaurav Chopra has a great paper showing from mouse models they're triglyceride rich lipid droplets. Another thing that might be going on here is that the lipid droplets, the stored lipids within the cells might be triglyceride, but more the secreted lipids could be different, could be cholesterol esters. So there could be like sub-compartment based as well. So this is an important and challenging question for the field. It's hard to tell at this moment.
Dave Holton from WashU. Michael, great work.
Thank you.
I just have a question, and I don't know how much you've addressed this, but in the in vivo situation, in some of the experiments we've done where we look for lipid staining with things like Bodippy in the brain of, let's say, mice that have amyloid in their brain or mice that have tau pathology and neurodegeneration, we don't see a big increase. We see some, but not a really big increase in neutral lipid staining in the presence of amyloid only, at least in mice. We see a huge increase in lipid accumulation in the tau model, especially when there's any neurodegeneration. It's boatloads bigger than what's in the amyloid models. But it doesn't seem like by co-staining it's in lipid droplets. It looks like it's almost all in lysosomes.
Sure.
So are you sure in the human brain samples that you looked at, are you sure that that oil red O staining is in lipid droplets, or could it be in other structures?
So we can't tell that from the oil red O staining. Great question. I think that there is a number of labs that have shown that at least in vitro, amyloid can induce these lipid droplets.
Yeah,
in vivo, I think there's a mixture of dying neurons and myelin debris, right, which is probably what's accumulating to that lysosomal lipid accumulation, and that probably is dominating the signal, I'd imagine, because there's probably a lot more of these exogenous lipids being phagocytosed. However, I think I could also be consistent with the endogenous production of these excess lipids. I think both are going on at the same time, and it's probably very difficult to disentangle the two.
Great, thanks.
Great talk as well. So, question about the co-culture experiment, or the experiment with I guess conditioned media. I have to be really close to that microphone. Almost I have to kiss that microphone to be heard. They good?
Yep.
Okay. What I was asking is whether in your co-culture experiments or your experiments with conditioned media coming from microglia, you had tested whether the neurotoxicity activity that you've seen requires some sort of internalization step in the neurons. In other words, do you think that you are poisoning lysosomes of your neurons with that condition? Or do you think it's through some other mechanism?
Yeah, I didn't have time to go into this here, but one of the ways I chose to investigate this is through these CRISPR screens in the neurons, seeing which genes are necessary for this toxicity. And the genes that we saw as being necessary, a lot of genes involved in uptake of lipids, trafficking to lysosomes, the degradation of lipids in lysosomes, and then finally genes involved in the transport of lipids into mitochondria. So this is something we're investigating right now in my lab, but to me that paints a picture of lipid transfer happening from the microglia to the neurons, very in a kind of a similar way to what you're suggesting, in that this transfer of lipids from glial cells in an APOE-dependent manner to neurons is causing this lysosomal or stress within the neurons that is then causing the downstream effects. So I think that's my working hypothesis at the moment.
That's great. Thank you.
I have one question. So do you have any idea relationship between amount of APOE and lipid drop formation? For example, if APOE4 is overexpressed, lipid droplet will be increased?
No, this is a really great question. I think there's really not enough known about how APOE regulates the intracellular lipid abundance. Sarah Cohen has done some really great work now showing that APOE actually localizes to lipid droplets and regulates lipid droplet size. I think there's something going on with the metabolism of the lipid droplet stores in APOE-dependent manner through mechanisms that are currently not understood at all. These are different than the trafficking and uptake mechanisms that APOE plays. So I think it's unknown, and it's a really important area of research to happen. But yeah, I can't answer the question at the moment.
The question online is, can the upregulation of ACSL1 be a result of lipid accumulation since it's also mediating fatty acid oxidation?
Yeah, so the regulation of ACSL1, I believe is...
The short answer, so we cannot draw any conclusion now because it's a two different model. We do also cross our E4 Christchurch or E3 Christchurch, which is the white-type human tau. We haven't done any analysis that will know. And the mechanistically, we don't know whether there will be the same mechanism, but based on the limited information, we think it could be different mechanism on that.
Thank you. So regarding this, do you think if you cross, sorry, I'm asking. So if you think APOE2 with Christchurch is the strongest protective APOE genotype, do you think?
You mean cross?
If the Christchurch induced APOE2 background, I assume this is a strong effect of anti-tau.
I'm sorry.
Yeah.
Oh, put together. So double. So we haven't done that. So the prediction, I mean, you think probably more protective, but we don't know.
Okay, so now that we can cross. Thank you, Yadong.
Thank you.
Okay, so next, the last speaker of today. So the next speaker is Dr. Yuan Chung from the Washington University.
Okay, shouldn't hold that point at the screen.
Okay, hi. So I'm the last one, and I guess everyone is tired. I'm tired too. But the good part is they already spoil everything. So yeah, we can go over this. So today I'm going to share the work on the biological effects of APOE3 Christchurch, specifically on the amyloid-induced tau-CDN spreading, and I will explain why. So we can quickly skip this because for the audience here, everybody knows these pathological classic simplified models where you have amyloid deposition occurs first, that initiates microglia astrocyte activation. At certain time points, the second phase of the Alzheimer's disease occurs with the tauopathy and then leads to the neuron degeneration. And then we know that from today's talk that APOE has been involved in all different aspects in terms of APOE4 as the most detrimental risk variant. And then as Dr. Yadong Huang mentioned, so about 5 years ago, so this is the first case report of a protective variant against the autosomal dominant Alzheimer's disease. And thanks for this unique individual, it has been reported that a unique patient who carries homozygous of APOE3 Christchurch mutation, it's R136S mutation, has been shown to its 25 years delay of cognitive decline, even though she carries the PCN1 mutation. So she's the only one has been found as a homozygous in her entire family. Now with the new data from the same group, they actually gather more heterozygous to show that APOE3 Christchurch protection can be also valid in those heterozygous with LASI-DAD. The most important thing is from the original study is, if you look at the PET scans of this particular individual, you'll notice that the amyloid deposition occurs in the same brain region where the rest of the PCN1 carriers has. However, if you look at the PHF tau burden in this unique individual, in those heavily amyloid deposit brain regions, there's almost limited tau pathology in this. That's what they mentioned too. If you actually draw a dashed line to actually separate this from what I did here, you can clearly see a separation between the amyloid pathology and the tau pathology only occurs in this individual. The rest of the PCN1 carriers, they clearly show the consistent overlapping of those brain pathological regions. So then we ask the question, why this happens? If the genetic evidence shows that she carries the homozygous mutation of this Christchurch, the first question we want to ask is whether this protection to separate amyloid pathology to tau pathology is actually real as genetic factors. If it's real, then how? How would that be possible? In order to answer that, we actually create the first humanized APOE3 Christchurch mouse, similar to the APOE3 mouse in our lab, and with the help from Taconic Bioscience. And then, sorry, a little bit back. Then we cross to the APP-PS1 mouse model. So the first evidence we see from the APOE3 Christchurch mouse is that we observe a peripheral hypolipidemia phenotype that has been also reported in that individual. You can see a clearly elevated plasma cholesterol and APOE levels. You also see the presence of the VLDL particles within this mouse that you usually do not see that in any black 6 mouse. However, this phenotype does not occur in the CSF and also the brain samples. We have more data analysis in the Western blot of the brain lysis, which will not be covered today. Then after we cross to the APP-PS1 mouse model, the first pathological evidence is we actually see a reduction, a mild reduction of amyloid pathology in this Christchurch mouse. You can clearly see a reduction of the amyloid plaque formation, which is also correlated to with the suppression of amyloid fibrillation in vitro. We have the data in the paper too. Then we perform this unique mouse model developed by the Virginia Lee's group at UPenn. We actually injected human tau extractions into the mouse brain and then assess the human tau seeding and spreading in this injection model. By injecting human tau extracts into the dentate gyrus and the hippocampus, you can clearly track the tau seeding and spreading into certain brain regions. More importantly, this mouse model can recapitulate amyloid-induced tauopathy, not only within the APA-positive tau aggregates within neurons, but also we can observe the near plaque tau pathology around every amyloid plaque. So this time, we actually observe a dramatic reduction of this amyloid-induced tau seeding and spreading in all different brain regions, including the primary injection site and also the secondary spreading sites. More importantly, we actually observe an even more significant reduction of near plaque tau pathology, specifically in this Christchurch mouse. You can clearly see that by AKA staining around the plaque, quantifying AKA staining around the plaques, you can see the dramatic reduction in the Christchurch mouse. And then this is also correlated with the reduction of the neuronal dystrophy around, sorry. Yeah, that's fine. It's also associated with the reduced neuronal dystrophy due to the reduction of the amyloid-induced tauopathy. So from previous literatures, including the studies in our lab, we actually show that the microglia activity around the plaque is essential to contribute to rescue the NP tau pathology in these mouse models, evidenced by the TREM2 knockout will actually disaggregate the NP tau pathology. So then we start to aim to assess the microglia specific around the plaques. This is also shown by spoiled by Dave. The first evidence we see that is we actually observed a consistently increased microglia clustering around each plaque. Also labeled by CD68, which is not only a stem microglia marker, but also a lysosome marker specifically expressed by the microglia. You can clearly see the elevated lysosome activity in those microglia around the plaques. This is quite important because this type of the improvement or the enhanced microglia activity is not only limited in the tau injection model. We also observe a similar improvement of microglia response in the pure amyloid mouse model without tau injection. We also see the similar increased activity in the brain regions where the tau has not spread yet. So on the right. Then we hypothesized the Christchurch mutation actually creates at least a unique feature in the myeloid cells carrying the Christchurch. And then we actually utilize the bone marrow-derived macrophage to mimic the myeloid cells, microglia in the brain. And we test the phagocytosis against the recombinant human tau fibrils. And also, the first thing surprised us is the A-beta fibrils. We do not observe a significant difference between the Christchurch macrophage versus E3 macrophage in phagocytosing A-beta fibrils. However, we do observe a specific increased phagocytosis of human tau fibrils in the Christchurch macrophages at different concentrations. In addition, the Christchurch macrophage myeloid cells can actually quickly degrade those human tau fibrils, even though they uptake in a very high level at the beginning. And then within 48 hours, they reach the similar level of the H3 macrophage after the phagocytosis. We also observed parallelly, so we also observed last tau seeds that has been secreted by the Christchurch macrophage, even though they uptake more tau fibrils in the beginning. So then comes with the more key question at the molecular level. Why does the APOE3 macrophage phagocytose more tau fibrils? This probably will be the most important mechanistic data experiment we perform in the paper. And so in addition to the human tau fibril phagocytosis experiment, we start to add lipidated human APOE3 and S3 Christchurch unlabeled together in the condition. So this is the same experiment as I showed just now. So at this concentration, you can see the elevated phagocytosis against the human tau fibrils by the Christchurch macrophage. If you add extra lipidated APOE3, you can actually see the suppression of the tau uptake, bringing Christchurch macrophage to phagocytose a similar level of as the E3 macrophage. However, if you add the same concentration of the lipidated APOE3 Christchurch particles, you actually do not see a statistically significant suppression of the tau fibril uptake. So together with other data, we also have more data in the paper. Due to time limit, I'm not going to go through everything. We have more than 100 panels. So from the beginning, we actually asked the question whether the genetic evidence of the Christchurch homologous can have the impact on the AD disease progression. We create mouse models. We observe the similar peripheral dyslipidemia as reported in the human case. We observe the less amyloid deposition, which also is consistent with the in vitro evidence. The Christchurch promotes a less A-beta fibrillization. More importantly, we actually show that the reduced NP tau pathology with enhanced microglia that is specifically observed in the Christchurch. So in addition, with cellular models, we demonstrate that all this phenotype is correlated with the enhanced phagocytosis and the reduced tau seeds. In the end, we actually show that all this enhanced phenotype is correlated with the protein reduction of the APOE binding to the HSPG and RP1. So I will stop here with all the, because only 10 minutes just passed. So I'm happy to take any questions.
Thank you. Any question? I'll start. So did you see the reduction of microglia in your Christchurch mastermind?
So we do see the reduction of the microgliosis due to the reduced pathology. But around each plaque, the microglia activity is enhanced.
Yeah, great talk. Mechanistically, how do you think that the Christchurch mutation would decrease the tau in the dystrophic neurites in the plaque? Because that tau is not from secreted or seeding. I mean, that is tau that is being stuck in the neurites because of lack of axonal transport and so on. So how does the microglia comes in contact with that tau and takes it away? Or is it that it's actually phagocytosing the whole neurite and getting rid of that? I mean, how do you explain that?
Yeah, that's a great question. So first part to exclude the possibility that microglia is actually phagocytosing the neurites, we do have the data to show by the synapsin staining and the PSD95 staining, we do not observe the reduction of that synaptic density around the plaques. We don't see that. We actually see a more protection in the tau injection model specifically. We don't see a change in the amyloid mouse model. Yeah. So, and then to directly answer that question, so we do not have the experimental evidence to show that microglia can phagocytose the secreted tau seeds, but that's what we hypothesize. So it's we tried several things, but it didn't work out. So it's very hard to monitor the actual tau seeds secreted in vivo. In vitro, you can actually reproduce that. Yeah. So yeah.
Hi, this is Mike Greicius from Stanford. I hate to do this. It's one of these. My question is the following statement, so you don't have to respond to any of this. But I just want to remind people that the Christchurch, the original paper obviously was in one individual, and the way that the genetic sort of filtering was done there was pretty atypical, in that there were a lot of potential protective variants that were left on the cutting room floor, both heterozygous and homozygous. Structural variants weren't looked at. So that's it's a very compelling hypothesis-generating initial paper. There's no question about that. The mouse work is gorgeous. Yudong's work, your work. I don't want to take away from that at all. But the human evidence, there's no statistical evidence currently that Christchurch is protected. The heterozygous paper that you mentioned, I think is a little controversial in that last year, Nick Cochran had a paper that also looked at the heterozygous and didn't show a protective effect. The ages at onset following some sort of clinical reassessment, which is not well defined in the New England Journal of Medicine paper, has been shifted rightward now. So, I just want to remind this group and encourage people to look at Nick Cochran's paper from last year, that there's still, in my view, no statistical evidence that Christchurch is protected.
Yeah, thank you.
Yeah, Dong Huang. Good question. So
yes.
I didn't remember in your paper, probably you mentioned the paper. Have you tried astrocyte? So you show a lot of microglia change in these contacts. So how about the astrocyte?
That's a great question. So I think partially included in the paper, we do assess the astrocyte around the amyloid plaques. We do not see a clear change in the astrocyte clustering around the plaques or this kind of behavior. We did not put the equal amount of effort to looking into the reactive astrocyte, these kind of markers. There's still a possibility that, because astrocyte also express certain receptors, HSPG, and also the LRP1. So it's still possible that astrocyte can get involved due to the same molecular mechanism. One thing, if you go to Seattle AD database, that you can actually see the microglia, the disease-associated microglia do upregulate HSPG, one type of HSPG, where you don't see that in the astrocytes. So this probably induced by the amyloid pathology that creates a different microenvironment. Yeah.
Thank you. Julia, TCW, Boston University, great work. Just going back, very simple about the APOE. Did you measure APOE level? Did you try to measure APOE level on the amyloid model versus tau model with Christchurch? They're decreasing or?
We did not compare the amyloid mouse model versus tau mouse model in this study. We do compare the APOE levels between the E3 and the E3 Christchurch without amyloid pathology. And we do not observe a significant difference in the CNS system.
So compared to E4, they're similar to Christchurch?
No, we did not do the E4. We were assessing the E3 versus Christchurch, E3 Christchurch.
They're similar.
They are very similar. However, in the peripheral, it's very clear that Christchurch leads to the dyslipidemia. So it cannot be cleaned by the LDR in the peripheral. So you see a very strong effect in the accumulation of Christchurch particles. They also start to form very large density particles. You can observe that by the EM.
So to comment on that, would you say it's similar to the APOE knockout mice in the liver, where they also have lipidemia kind of phenotype?
Depending on how you define the phenotype of hyperlipidemia, if you only rely on the cholesterol level and the triglyceride level to define that, maybe they are similar. But in terms of the mechanism, we think that it's very close to the APOE2. Because APOE knockout does not contain the HDL containing APOE, right? The HDL will not be formed by the APOE.
Just add on a comment about the APOE level. So we see very similar in our E4 case. If you look at peripheral, there's an accumulation of this Christchurch APOE because there's impaired clearance or receptor binding. If you look at the brain, actually E4 tau and Christchurch E4 tau has some of the level. Seems in the brain they are very similar. We've also seen the culture iPS cell somehow this Christchurch E4 also accumulates in the culture. We don't understand why is that the case, but in the brain they both.
Yeah, we see the very similar things. Yeah.
All right. Thank you very much. So now that we can close this session, thank you, everyone.
Okay. I was going to ask you, Dwayne, to load that 100 slides that we have to. All right, so we're going to do a short wrap up. I'm going to do the genetics, and David's going to do everything else. So I would say our first talk was discovery to diversity, and for the young people, this was in 1993. Technology was not like it is today. It just shows that, if you're observant and you have good ideas and you listen and you put things together, you don't need great technology to make a major contribution. And also, what's old is new in terms of the risk for APOE4. It's like watching a remake of a movie, maybe. Diversity gives us a lot of tools we saw to look at APOE4, and I'll remind you that Christchurch, whether it's real or not real, thank you, Dr. Greicius. It was found in South America, and so it just shows the usefulness. I think genetic studies support lowering expression as correlated with risk, and I think we've seen that. Also, we also found today that Lindsay Farr and Francis Collins were both right about, Lindsay will go to bed sleeping well tonight. And it may be evolved with R loop and the enhancer from the R loop. So that's interesting, I thought. We also saw that there are haplotypes probably that differ between APOE3 and APOE4. So while we concentrate on the protein, let's remember that the haplotypes around it may be well contributing. Finally, we saw that protective loci, they have generations to work out through nature that what works for protection, but doesn't necessarily give a lot of side effects. And so they're useful things, and I think we should continue to really focus on protective variants as well as those that give risk. We think that 50% of APOE is probably okay, and we definitely need to continue our work on APOE2, I think.
Thanks, Jeff. I'll just make a few comments about the mechanisms sessions. So I think for both Liwei, Allison, Julia, and Chris, what their really exciting studies really show is that on a variety of cell types in the central nervous system, whether it be oligodendrocytes, astrocytes, microglia, and in some sense neurons, that APOE can have effects on these cells that affect different pathways. Some of it could be non-cell autonomous effects and some cell autonomous. And I think which of these effects that they're having on those cell types is relevant for disease pathogenesis in vivo, I think will be really important to sort out as we go forward. But clearly, there's really interesting effects on lipid metabolism in the cells, the matrixome pathway, the inflammatory pathways, depending on which cell type. And I think this really exciting data is going to move forward, and we'll have to sort out what's most relevant, especially from a therapeutic perspective on looking at APOE's effects. I think Gil DiPaolo's talk and Michael Haney's talk both emphasize in different ways the very important role of lipid handling by both microglia for sure, and also probably neurons, and how APOE isoforms can alter that lipid handling in different ways. And I think that's going to be something very important to sort out, not just mechanistically, but how you handle that lipid from a therapeutic perspective may be another way to attack APOE in novel ways that really hasn't happened yet in people yet. And then both Yadong's talk and Yin's talk, whether or not in humans the Christchurch variant really is showing this protective effect in that person, I think there's I think probably it is, but we'll see. But I think the animal data, I've never seen any data like this before, where a single change, where the only thing you're changing is one variant in one protein in an animal, and you see these dramatic protective effects against, in one case, amyloid-induced tau seeding and spreading, and another one against tau-mediated damage. That's something to pursue. Whether the human case is correct or not, I mean, I've never seen anything like that. That's something I think we have to figure out what's going on there, because that offers a real way into therapy, I think. And so I'll end on that positive note, because I think there's lots more to come tomorrow about this. So, thanks.